Please turn off your device camera. Participants are muted by default to enable the speakers to present without interruption. Participants may use the question and answer chat box to raise their questions. They should first state their name, affiliation, their question, and the speaker they want to answer. To get your certificate of attendance from this webinar, participants are asked to complete the evaluation form. The link will be posted in the chat box at the end of the webinar.
Tayo po ay magsitayo. Bilang pagpupugay, ilagay ang kanang kamay sa ating dibdib at awitin ang pambansang awit ng Pilipinas.
Ladies and gentlemen, your host, Dr. Alayla Dicadis.
Good morning, everyone. So this is the current biochemistry, molecular biology, biotechnology, and researches and services in Mindanao. This activity is co-hosted by PGC Mindanao and co-sponsored by DOST Region 11. This webinar, as the title suggests, aims to showcase current biochemistry, molecular biology, and biotechnology researches and services in different institutions in Mindanao, from Central Mindanao University, University of Southern Mindanao, in the banana industry, Tagum Agricultural Development Company Incorporated, or TADECO, Mindanao State University, General Santos City, and the Philippine Genome Center, Mindanao, here in UP Mindanao. After every 4 presentations, there will be an open forum, and we will be joined later by Dr. Lair Ani Morao as our moderator. Please be reminded of our house rules for this webinar, which was played before the start of the program. Here's the flow of this morning program. Now, to officially start our program, let's hear a welcoming message from Dr. Annabel Novero, Professor 6 and UP Scientist 1 of the Department of Biological Sciences and Environmental Studies, College of Science and Mathematics, UP Mindanao, and the current president of PSBMB Mindanao chapter. Let us welcome Dr. Annabel Novero. This is to be followed by the opening remarks from the Regional Director of DOST Region 11, Dr. Anthony Sales.
Dr. Anthony Sales, Regional Director, DOST 11, Dr. Kenneth Baroga, Assistant Regional Director for R&D and Innovation, DOST 11, Dr. Lair Ani Morao, Program Director, Philippine Genome Center, Mindanao, Dr. Leslie Dalmasio, President, Philippine Society for Biochemistry and Molecular Biology, distinguished guest speakers, fellow officers, and members of the PSBMB, participants, good morning. We are absolutely thrilled to present to you this half-day webinar as a sort of a revival activity of the PSBMB Mindanao. We have last come together in October 2017 in General Santos City for the first Mindanao Biochemistry and Molecular Biology Conference. A lot of new and exciting research have come up since then, and today we will hear about some of these endeavors. I believe that the pandemic has forced the layman to be familiar with the common molecular biology terminologies like RT-PCR, DNA, and mRNA. It is our responsibility as scientists to educate and not alienate the public on the powers of science in order to help them make informed decisions on matters affecting daily life in the pandemic. An Omics webinar is slated in October to celebrate the third anniversary of PGC Mindanao. The PSBMB Mindanao is honored to return this favor by co-hosting that exciting event. Linus Pauling, the recipient of the Nobel Prize in Chemistry in 1954, said in an interview, and I quote, "I've tried to fit knowledge that I acquired into my understanding of the world, and much of the new information that I learned, new discoveries that have been made, seem to me to be compatible with my existing understanding of the world. When something comes along that I do not understand, I can't fit in. That bothers me. I think about it, mull over it, and perhaps ultimately do some work with it. I was able to make discoveries in molecular biology in the early days when the subject didn't exist." Unquote. So today, it is my hope that you will find something that you do not know and that it will bother you enough to make you want to do something about it for the good of our country. And with that note, I welcome you all to this webinar. May we have a productive day. Daghang salamat.
Maayong buntag. My warmest welcome to our esteemed guests and participants who are here today for the webinar hosted by the Philippine Society of Biochemistry and Molecular Biology, Mindanao Chapter, and co-hosted by the Department of Science and Technology 11 and the Philippine Genome Center, Mindanao. It is already 2021. We are only 9 years away from the supposed completion of the Agenda 2030 on the Sustainable Development Goals, or SDGs, which is our lifetime opportunity to prepare the future generation for resiliency through evidence-based, inclusive implementation of science-based solutions. What have we done so far? What should we be doing? It is with an exciting and proud Mindanaoan heart that I am opening this webinar along with the key implementers among our Pentahelix collaborators from the government sector, academic sector, industry sector, the civil society, and the media. We shall be showcasing some of the current researches, services, projects, programs, and initiatives in the disciplines of biochemistry, molecular biology, and biotechnology, establishing a culture of scientific research, development, and innovation, which translate outputs to support the universal goal of the Agenda 2030. Regardless which sector you represent, it is our collective mission to inspire and transform communities in employing scientific, engineering, technological, and innovative solutions to address current and emerging challenges and realities in Mindanao. I hope that this webinar will continue to bring inspiration to future scientists and to fuel higher aspirations to persevering researchers and innovators of today. May we all have a scientific and productive day. Once again, maayong buntag sa inyong tanan.
Thank you very much, Dr. Novero and Dr. Sales. Now let's get this started. To start, allow me to introduce our first speaker. Dr. Reggie de la Cruz is a Professor 5 of the Department of Biology, College of Arts and Sciences, Central Mindanao University, Muswan, Bukidnon, from 1996 to present. She is also the program head of CMU Tuklas Tunas Development Center, a DIAD Simeo Cerca Fellow, Vice President of PSBMB Mindanao Chapter, President of Biota Bukidnon Chapter, and former President of the Philippine Society for the Advancement of Genetics. She was the NAST Biotechnology Focal Person for Region 10 in 2013, and she was also the NAST Outstanding Scientific Publication Awardee in 2018. She has cloned and sequenced a total of 20 genes in coconut and macapuno and has authored several ISI publications. To discuss the current biochemistry, molecular biology, and biotechnology researches and services at Central Mindanao University, let us all welcome Dr. Reggie de la Cruz.
A pleasant day to all. It is my honor to present to you the current molecular biology, biotechnology, and biochemistry researches in Central Mindanao University, CMU, Muswan, Bukidnon. CMU is ISO 9001:2015 certified, has a 3QS star rating, and ranked 9th in the Philippines according to the Webometrics ranking of world universities. CMU is CHED Center of Excellence in Agriculture, Biology, Forestry, and Veterinary Medicine. CHED Center of Development in Mathematics and Teacher Education. AACOP Level 4 Phase 2 re-accredited for the BS Bio program. DOST and CHED Delivering Institution for MS and PhD Biology. Our university has a beautiful campus with forests all around, making it conducive for learning, and as such, it is tagged an academic paradise. Our very first update is that the Department of Biology now offers the Molecular Biology and Biotechnology track for our BS Biology, MS Biology, and PhD Biology. It has been 7 years since MBB researches took off in CMU. Now we have done the following: PCR-based gene cloning, molecular identification, transcript profiling, molecular diagnostics, and protein fingerprinting. Several of these researches came about when CMU was awarded Tuklas Lunas Development Center in 2014. By then, we ventured into drug discovery, chemical profiling, and isolation of bioactive compounds. The number of biotechnology researches also increased through the years. We are on to mammalian cell culture, tissue culture on banana orchids, spore culture of ferns, mushroom culture, functional food development, biopesticide development, and crop natural selection. For the CMU Tuklas Lunas Development Center, its pilot project was exploring the potentials of the Philippine ferns and lycopods as therapeutics for chronic inflammation and cancer. This project paved the way for equipment upgrading and the capability building of all the researchers. The said project has 4 studies and has a funding of 29 million pesos. Inflammation has long been the focus of CMU Tuklas Lunas Development Center. Being a silent killer, inflammation, if undetected for a long time, will cause diabetes, cardiovascular disease, Alzheimer's disease, allergy, and even several forms of cancers. Thus, there is a need to develop drugs to fight inflammation. The said project screened for 28 species of ferns and lycopods, and finally arrived at 3 extracts which are cyclooxygenase inhibitors and non-toxic to skin, kidney, liver, and heart cells. The identified 3 extracts are now into product development. Another highlight of the project was the isolation of 3 bioactive compounds. Phase 2 of CMU TLDC is now on a program entitled PharmaFern Development of Health Products from Ferns. PharmaFern took off in 2019 and has 5 projects. The first project is on the establishment of a terra nursery for the mass propagation of selected ferns led by Dr. Glenda Doblas. The second project is on the development of functional food from the said ferns, and this was headed by Dr. Jose S. Balmurida. In collaboration with the College of Pharmacy of UP Manila, specifically through Professor Jocelyn Palakpak and Professor Ethel Ladignon, we have formulated an anti-inflammatory ointment from the identified 3 species from the Phase 1. Patent applications are now being drafted. Project 4, led by Dr. Melania Inot, is in charge of chemical profiling and standardization of the herbal products, while Project 5 is into isolation of anti-inflammatory compounds from the selected ferns. This is headed by Eileen May G. Ang, previously, and now by Dr. Gina B. Barbosa, in collaboration with the UP Diliman Institute of Chemistry, Professor and Dr. Irene Villaseñor. To date, PharmaFern has filed for 4 utility models and 3 patents on functional foods, established a 1-hectare fern production area, 2 greenhouses, an animal house for CMU counterpart, and the first HPTLC standardization laboratory. It also has collaborations with the UP Manila College of Pharmacy, UP Diliman Institute of Chemistry, and the University of Melbourne, Australia. In the near future, we will be catering to all Mindanao researchers for the following: cytotoxicity assays using human skin cells, HEC-N, enzyme-based assays on COX and LOX, animal-based assay, particularly toxicity and paw edema assay. Aside from the Tuklas Lunas Development Center researches, one of our favorite are the Makapuno gene discovery studies by our students. To date, more than 10 partial genes have been cloned and sequenced, and one of our papers bagged the 2018 NAS Outstanding Scientific Publication Award. The paper reported mutations in the alpha-D galactosidase gene which suggests molecular basis of the mutant makapuno coconut phenomenon. To support biodiversity researches, which is one of the strengths of CMU, we have several studies on DNA barcoding, especially for difficult to identify species. To highlight a few, we have this study by Elsa May de Lima Baron on the diversity of anurans and systematics of Philippine bush frogs Philautus from select mountain ecosystems of Mindanao Island, Philippines. A total of 30 anuran species were studied, and there is one potential new species discovered. 5 of our DUST Strand scholars, 1 PhD and 4 MS, worked on DNA barcoding and just recently graduated on time. To highlight their studies, we have here first by Dr. Vanessa L. Kalimbo on the bacterial characterization and in vitro antifungal potential of cutaneous isolates from selected Philippine endemic frogs. Several of her bacterial isolates have inhibited the growth of Candida tropicalis and Aspergillus niger. Another interesting study is by Michelle Suelo on hawk moth species, the Sphingidae. She was able to generate several new DNA barcodes. Another DOST strand scholar in the person of Jade Rosas was able to generate also the barcodes of the species of Phasmatodea in Muswan, Maramag, Bukidnon. Rona May Viernes was able to generate also the barcodes of the giant silkworm moths in Bukidnon, Philippines. Another study is by Gentian Alandergantes on spiders. 13 species were identified, and there is a new distribution record of a certain species in Southeast Asia, and with 3 new polymorphs reported. A very important application of molecular biology is in the molecular detection of plant pathogens. We have several students who did this, and many of them are from the Department of Pathology of the College of Agriculture of CMU. First, an MS biology student, in collaboration with Unifruti, worked on the detection of Fusarium oxysporum sp cubense in banana using qPCR and was able to detect FOCTR4 rays in 2019. To highlight some of the studies we have by Carla June Siadao in 2019, who used 4 types of primers to detect Fusarium oxysporum FSP lycopersicide and was able to detect race 1 and race 3 in selected tomato farms in Dalwangan, Bukidnon. Chris Joy Ranalan also successfully detected Santomonas uvisicatoria in pepper infected for bacterial leaf spot. Of the 5 area studies, only 1 was found to be positive. If you hear the term biotechnology, you would think that it would need special facilities and laboratories. But for us, we are happy, as a Department of Biology Extension, to share the technology on backyard spore culture of staghorn fern to women as an alternative livelihood and as a help to biodiversity conservation. So let's now have a glimpse on the laboratories and facilities of Central Mindanao University doing molecular biology biotechnology, and biochemistry work. Then we have the Natural Science Research Center. In the Natural Science Research Center, we have the Plant Spore and Tissue Culture Laboratory. Then we have the Natural Products Research and Development Center. The College of Agriculture has also their Tissue Culture Laboratory. The CMU Village Level Biocon Laboratory is for biological controls. Several services are available in our Soil and Plant Analysis Laboratory, or SPAL. We are also proud of our CMU Mushroom Center. The College of Veterinary Medicine has its Animal Disease Diagnostic Laboratory. To emphasize on our future and new services in the university,
first is the TLDC Mammalian Cell Culture Lab that will be able to accept cytotoxicity assay using human epidermal keratinocytes, HEC-N. Then we have the TLDC Animal Assay Lab, able to cater poedema and toxicity assays.
there is still much in store for Central Mindanao University. Thank you very much for listening. And to God be the glory. Special mention to the people behind the Tuklas Lunas Development Center, especially to my team.
Thank you very much, Dr. Reggie. I hope you continue mentoring our students, no, with your various available researches in CMU. You may type in your question for Dr. Reggie de la Cruz in the Q&A box, and then we will read them later during the open forum. Okay, our second speaker is Dr. Edward A. Barlaan. He is currently working at the University of Southern Mindanao as Professor 6 and Vice President for Research, Development, and Extension. He is formerly affiliated with the Institute of Biology, UP Diliman, National Institute of Crop Science, Japan, and International Rice Research Institute. He took his PhD in agriculture in Ehime University, Japan, specializing in plant breeding. His other fields of specialization and interest include genetics, agronomy, biology, cell and molecular biology, molecular diagnostics, microbial ecology, and plant pathology. He published more than a dozen of articles in ISI Index international journals with over 300 citations. He received research grants from various funding agencies with a consolidated amount of more than 120 million pesos for research projects in milkfish, coconut, oil palm, cacao, banana, and mango. He has filed 3 patents and 11 utility models. He is a recipient of awards such as Civil Service Commission Pag-asa Award for Rice Improvement, Best Paper in Research category in BA Bar, and second Best Paper in Research category in BST Picard, Outstanding Alumni Award, and Invention Disclosure Award, among others. So to discuss the current biochemistry, molecular biology, and biotechnology researches and services at University of Southern Mindanao, let us welcome Dr. Edward Barlaan.
Good morning, everyone. Thank you very much for making time to join the webinar today. This morning, I will be sharing to you the current biochemistry, molecular biology, and biotechnology researches and services in University of Southern Mindanao, or USM. For those who are new, you may be asking, where is USM? Well, of course, it is found in Mindanao, particularly in Cotabato region, and specifically in Cabacan. University of Southern Mindanao is one of the 4 leading state universities in the Philippines as the center of excellence in agriculture. It is also the center of development in biology and veterinary medicine. It is one of the 9 among 120 SUCs with level 4 status, and it is an ISO certified university. USM has a total area of 5,129 hectares. Specifically in Cabacan main campus, it has 1,024 hectares. In Arakan Valley campus, it has 4,091 hectares. In Cadapawan City campus, it has 14.9 hectares. For more details about USM, please visit the website on www.usm.edu.ph. I will be sharing to you the biochemistry, molecular biology, and biotechnology researches in USM, particularly the new, the ongoing, and recently completed projects, and I'll be sharing to you by commodity. First, we have the cacao. We have a newly funded and implemented program, the Niche Center in the region, or the NICER, Cacao R&D Center, which has 3 project components. The project 1 is the molecular fingerprinting of cacao parental recommended HYVs and through Creole U, ensuring multiplication of quality planting materials, or QPMs, for increased productivity and profitability. This project makes use of the SSR markers or molecular markers for fingerprinting of the NSEC recommended high-yielding varieties, as well as the true Creole U cacao parental or mother trees for multiplication of quality planting materials. And project 2 is the upgrading of cacao gene bank for conservation and management in cacao varietal improvement. This project will enhance the germplasm collection in USM that will be used for cacao breeding, particularly in the utilization of marker-assisted selection or breeding. For Project 3, this is the development of optimized post-harvest processing approaches for improved quality of cacao beans. We will be optimizing or standardizing the post-harvest processes in cacao, and this in order to generate improved bean quality, and these beans will be processed or analyzed using the different compounds associated with bean quality. And another project is the validation of molecular markers for identification of cacao HYVs, Creole types, and disease-resistant varieties through marker-assisted breeding. Uh, is somewhat similar to the previous one, yet this emphasizes or focuses on the seedling materials in different nurseries in cacao uh, regions or in nurseries in different regions, particularly on the identification of HYBs using molecular markers, as well as the Creole types using the SR markers, and also the identification of disease-resistant varieties using those SR markers. Another project, the newly completed one, is the functional genomics assisted development of gene markers for economically important traits in cacao production and varietal improvement, in which this project was able to generate SSR markers that can fingerprint the insect recommended cacao varieties specifically, and also sets of SSR markers that can identify the true Creole types, as well as the SSR markers that are associated with resistance to major diseases in cacao. Another commodity is the mango. This is the Mecoteco, or this is a joint project between Philippines and Taiwan on the improvement of Carabao mango production and in fruit quality through gene identification for scab and stem and rot resistance by genome-wide association studies. Um, the The project aims to assess the Carabao mango strains for resistance to scab and stem and rot, to generate GBS database of Philippine mango germplasm for scab and stem resistance, to develop molecular markers using GBS and GWAS for scab and stem in mango, and to validate molecular markers for utility in marker-aided selection. Another project is the fruit quality improvement in Carabao mango through identification of genes for scab and stem and rot resistance by genotyping by sequencing or GBS and genome-wide association studies or GWAS. This has similarity with the previous one, but the previous one focuses on the Carabao mango, and this project focuses on the non-Carabao mango. So this project aims to evaluate the Philippine non-Carabao mango varieties and strains for scab and stem and rot resistance, to establish GBS-generated sequence database for stem and rot and scab resistance, to generate molecular markers for scab resistance using GBS and GWAS, to validate molecular markers for utility in marker-assisted selection. Another project in mango is the pilot testing and validation of SSR marker kit for Philippine mango germplasm in commercial mango nurseries. This is a completed one. And this project was able to analyze the phylogenetic relationship among Carabao as well as the non-Carabao mangoes. We have another commodity, the rubber. This is newly funded and newly implemented. The screening of potential endophytes as biocontrol agent against major and emerging leaf diseases of rubber. And this project aims to determine the occurrence and prevalence of major leaf diseases as well as emerging leaf diseases in rubber, to determine the diversity of endophytic fungi of rubber collected from major plantation areas, and to evaluate the antagonistic activity and possible mode of action of endophytic fungi against major and emerging leaf diseases of rubber, to identify effective endophytes through combined morphocultural and molecular techniques, and to evaluate the efficiency of colonization of endophytes and recovery from inoculated rubber plant to verify endophyty. Then we have another newly completed project in rubber, the genomics assisted development of gene markers for important traits in rubber production and clone improvement. This project was able to generate markers from two approaches, the gene mining and the genome filtering. They have generated 4 molecular markers that are associated with resistance to Phytophthora palmivora, and these markers can be used for identification and selection of resistant clones against this pathogen. And we have another commodity, the banana, the newly completed one. This is the molecular identification and detection of microbial pathogens of banana. And this project was able to generate products and technologies that can help the banana industry, particularly on the quantitative real-time or qPCR detection of Fusarium in banana plants. You can see in here the infected ones and the healthy ones. Not only that, we have the technology using the digital PCR detection of Fusarium. And as you can notice in here, the infected ones with high blue, and they are healthy. They are healthy, yet they may appear healthy, but you can notice in here the dots here in which they are quantified. So they are visually healthy, yet molecularly infected. So we have also for banana bunchy top virus, and we're able to have the primers, generated primers that can identify using the endpoint PCR identification of BBTV in infected plants from different sources. And we have also the qPCR detection of BBTV in different banana varieties. And furthermore, we have the digital PCR detection and monitoring of BBTV in banana. About bacterial health, we were able to develop qPCR detection of Ralstonia solanacearum in banana, as well as the digital detection and monitoring of solanacearum in banana. So we have another project in banana, the banana floral apex tissue culture, and which is being pitched now. And this micro propagation technique is more efficient, particularly in developing the pest disease-free banana. Start using the starting material, the floral apex. Then we have another commodity, the oil palm and palm species. This is newly completed. This is the identification and molecular profiling of oil palm varieties and hybrids towards oil palm variety improvement in the Philippines. This project was able to generate phylogenetic trees and that can differentiate palm species in the Philippines, as well as their different oil palm varieties. In fact, it was able to generate here fingerprints of Philippine palm species using the developed SSR markers. These are the samples of this. Then we have another commodity, coffee, the newly completed one, the phenotypic and genotypic analysis of Coffea canifera or the Robusta using the morphological and molecular markers. This project was able to have inventory, documentation, collection of samples, and geotagging of commercial Robusta coffee nurseries in Socsargen, Davao Region, Northern Mindanao, Oriental Mindoro, and Benguet. And they have visited 45 nurseries and have used 64 SSR primers and evaluated generating 16 functional primers that was used in the study. And we have also studied our project in rice, the harnessing insect bat ecosystem services for economical rice production. This is just newly completed. And there are several components of the study, and I will be featuring in here the relevant one, particularly on the bacterial quality and chemical content of insect bat guano as organic soil amendment. So they have analyzed in here the bacterial density, as well as the pH, the moisture content, the nitrogen, the organic matter, the phosphorus, and specifically the abundance of bacterial genera and bacterial species in guano. Then another commodity is the durian. This newly completed one, the pilot testing and validation of the molecular marker diagnostic kit for durian nurseries in Mindanao and other parts of the Philippines. And this project was able to generate genetic profile of different durian varieties and clones using SSR markers. We have also project in vegetables, particularly the performance and evaluation of fermented teas as biofertilizer and biopesticides, and their effects on pest, diseases, and yields of selected solanaceous vegetables, in which they have used different raw materials and they have fermented this one, and they wanted to determine if this tea product can be used as biopesticides or biofertilizers for organic solanaceous vegetable. And we have other plants, particularly on the exploration and evaluation of indigenous plants in North Cotabato with pro and anti-angiogenic properties. And This is newly completed, and this project was able to identify compounds and promising indigenous plants, having analyzed the different compounds that are associated with anti-cancer property. And likewise, there is a Toklas Lunas program, this project in here, the screening of plants from Region 12 with potential anti-cancer activity. So this project aims to identify bioactive plant extracts with anti-proliferative and anti-migratory potential against selected cancer cell lines. Also, to evaluate the toxicity profile of bioactive plant extracts, the nephrotoxicity, hepatotoxicity, and cardiotoxicity, and to confirm the anti-proliferative and anti-migratory potential of non-toxic bioactive plant extracts through relevant orthogonal assays. Then we have the human and macaque interface. That is the prevalence and the molecular characterization of gastrointestinal parasites in human macaque interface. The case of Hindang Leyte and New Israel Makilala, North Cotabato. Actually, this is a collaboration with UP-Mindanao, UP Science, and Visayas State University for the program Human Macaque Interface, or the HUMANE. Now I'll be sharing to you the biochemistry, molecular biology, and biotechnology services in USM. We have the central laboratory, in which it focuses on agriculture-related analysis, particularly the chemical test for plant tissue, food, feed, water, and phytochemical analysis. We have also the physical and the chemical test for soil and chemical test for fertilizers. We have also the USM College of Science and Mathematics, Bio Department, Microbiological and Chemical Testing Laboratory, which focuses on water potability testing based on bacteriological and physical analysis. They have also this food microbial analysis with a total plate count, aerobic plate count, mold and yeast count, total coliform count, fecal coliform count, with these pathogens such as Salmonella aureus, E. coli, Staphylococcus aureus, and other foodborne pathogens. And they have also microbial cultures that that's used for academic and research purposes. Then we have the biotechnology, particularly the services for tissue culture. We have services for tissue culture in Lakatan, Cavendish, Cardava, Abaca, as well as they offer tissue culture training in limited capacity. Then we have the molecular diagnostics laboratory that can identify the plant variety molecularly, whether by fingerprinting, forensics. And this can be done in rubber, cacao, coconut, and durian. We have also pathogen detection, indexing, and monitoring of these particular pathogens, the BBTB, Fusarium oxysporum, Ralstonia solanacearum, and also other services like microbial molecular identification. And we also offer molecular techniques training in limited capacity as well. So in summary, for the biochemistry, molecular biology, and biotechnologies researches and services in USM, we have researchers, a total of 27, with new 6, ongoing 4, newly completed 17, and for the services we have 10. And for the external funds, we have the UST Picard, the PCHRD, PSHIRT, DA for Department of Agriculture, the DA Bar, the DA Biotech, and we have the CHED. And so, thank you very much for listening.
Thank you very much, Sir Ed. Drooling kami dito. Ang dami nilang projects, no? So sana all ang ISO services. Thank you for your presentation, Dr. Ed Barlaan. Now let's move on to our third speaker. Our third speaker is a research manager of Tagum Agricultural Development Company, or TADECO, where she spearheaded the TADECO Disease Diagnostics and Services Laboratory and Post-Harvest R&D Department. She led several projects, mostly on method development and validation focusing on the management of plant diseases, especially those affecting Cavendish bananas. One of the highlights of her career is the establishment of TDDS Laboratory as the first plant pathology laboratory in the Philippines to be accredited to PNS ISO IEC 17205-2017. Her extensive research experience in laboratory and field conditions allowed her to incorporate molecular tools in field disease management. She is passionate about research and enjoys working on projects related to host pathogen interaction, fungicide resistance, diagnostics, and disease control. She holds a master's degree in plant pathology with a cognate in plant physiology from the University of the Philippines in Los Baños, where she also earned her bachelor's degree. Bea, as fondly called by her friends, is currently pursuing her PhD in plant pathology at North Dakota State University, USA, specializing in the genomics of Fusarium graminearum in barley. So to discuss the current biochemistry, molecular biology, and biotechnology researches and services in the banana industry, let us welcome Ms. Abea May Navasca.
Good morning, everyone. I hope you are all doing very well. It has been truly an honor to be invited by the Philippine Society of Biochemistry and Molecular Biology for this webinar. I am Abea May Navasca from Tagum Agricultural Development Company, a producer of premium and export-grade Cavendish bananas. And for this morning, I will be sharing with you the current biochemistry, molecular biology, and biotechnology researches and services in the banana industry. And I do hope that at the end of this presentation, you will be more interested in our researches and that you will be availing our services. At this point, you might be wondering why the banana industry was given a particular slot for this webinar. And as you may probably know, bananas are big in Mindanao, especially in the Davao region. In the recent quarterly update by the Philippine Statistics Authority on major fruit crops, Davao region accounts for 36% banana production in the Philippines, reaching to almost 750,000 metric tons, with the highest production coming from the export variety, Cavendish at 55.5%, followed by Sabah, Lakatan, and others. Here I am showing one of the processing stations of Tadeco where we prepare banana fruits for export. On the average, Tadeco contributes 30 million boxes of 13.5 kilogram bags annually. We produce fruits following the highest standards as Tadeco is Global GAP certified and the only certified banana company that received international award on Global GAP. Sustaining an export grade, high productivity every year requires a strong research support to innovate and validate disease management procedures. And it is in this mindset that Tadeco invested in a state-of-the-art laboratory which led to the launching of the Tadeco Agro-Technology Outreach Program. This program is a means to offer our services to everyone, especially those in the banana industry. This program was launched on March 26, 2018, where we specifically constructed a satellite receiving facility for convenient submission of samples. We have assigned one colleague to cater inquiries and orders in that facility. We are serving our customers 6 days a week, Monday through Saturday. And what is really nice about this program is that the services that we offer to our customers are the same services that we use in Tadeco. Apart from the laboratory services, we are also giving free seminars to our customers. This is in 2019, and we have actually planned for 2020 but have to cancel because of COVID-19. The decision to offer our services comes with our competence. the Tadeco Disease Diagnostics and Services Laboratory is the first and by far the only plant pathology laboratory in the Philippines to be accredited to PNS ISO IEC 17025-2017 standards. With this accreditation comes our expertise in method development and method validation equipped with the rigid trainings and education, suitable tools, and high-end equipment. Our chemistry laboratory is compliant to the provisions of Republic Act 10657, an act regulating and modernizing the practice of chemistry in the Philippines. And our banana tissue culture is the very first banana tissue culture laboratory in the Philippines to be accredited by the Bureau of Plant Industry. We also take pride in receiving trainings here and abroad from the Philippine Genome Center in UP Diliman, Promega Singapore, Biorad Singapore, and Forestry and Agricultural Biotechnology Institute in South Africa. We value collaborations from our public and private partners in doing research. Currently, we have a project with Cagayan State University on the development of efficient semiochemical management method for banana flower thrips with Dr. Cecilia Reyes. This is through Cradle Project of the OST. And we also have a partnership with Mindanao State University, General Santos City, with Dr. Lynn Ralios on the molecular and morphocultural analysis of black Sigatoka pathogen populations in banana grown in Mindanao. And in the past, we have also taken international collaborations with Wageningen University and clear detections from the Netherlands on the Promo Banana Project. In the future, we look forward to collaborate with Dr. Lailani Dacones of UP Diliman for fungicide resistance and, of course, genomics projects with PGC Mindanao. One of our current projects with our private partners is on the optimization of the real-time PCR detection of banana brack mosaic virus. This is with Promega, and this is in line with the real-time PCR grant that we obtained in 2019. There were 3 grants, and Tadeco was chosen as one of those grants. And apart from detection projects, we also do gene expression analysis, one of which is with STK Biotechnologies on the enzymatic assays and gene expression analysis on the activation of defense mechanisms by tea tree oil against Fusarium tropical race 4 in Cavendish bananas. And this is a pioneering study in the Philippines. Apart from these partnerships, a big part of our researches is geared toward effective, efficient, and sustainable disease control by doing screening and efficacy tests. We also offer these services. This is in collaboration with agrochemical companies and even with our internal research teams. Black Sigatoka and Fusarium wilt are the top 2 in our priority list. Black Sigatoka, especially with its growing concerns on decreasing sensitivity and, of course, our continuous battle with Fusarium wilt. We also do researches and research services on insect pests, nematodes, and weeds. In terms of laboratory services, we offer this commercially, the molecular detection of TR4 in plant tissues, water, and soil. This is very important, especially in monitoring procedures as basis for management decisions. We are also offering molecular detection of the banana bunchitap virus and Candidatus asiaticus liberibacter, the one causing citrus greening. And as mentioned, we are currently optimizing and validating the detection of banana brack mosaic virus with Promega, which we hope to offer soon. Being in an agricultural company, our services are geared toward agricultural pests. Our entomology and nematology laboratory is equipped to do root health system determination, nematode and insect identification, nematode population determination, and even provision of Rhodopholos similis cultures. In the hands of competent and licensed chemists and chemical technicians, we can do the following tests: macro and micro element determination in soil, leaf, water, and fertilizers, heavy metals analysis, humic acid analysis, organic matter and soil texture, physico-chemical analysis of water, active ingredient in pesticide formulation, and dry matter determination in fruits. We also offer disease-free planting materials. As mentioned, our tissue culture laboratory is the very first banana tissue culture laboratory in the Philippines to be accredited by the Bureau of Plant Industry. And this laboratory is also complemented with a semi-automated nursery equipped with modern technology. And since we have high demands for this, we schedule the orders of our customers. The plantlets that we offer are treated with trichoderma. This is in line with our approach in the management of fusarium wilt, where we use biological control agents such as Trichoderma asperellum. I'm showing here a classic dual culture assay of Trichoderma asperellum against TR4, simulating conditions where trichoderma is applied as preventive or curative treatment, as well as conditions placing both fungus at one time. And we have validated the effect of trichoderma as preventive approach in the field, and we have been using it for years. And we offer this product commercially, either in oatmeal or cracked corn form. We are continuously developing other biological control agents, which we hope to offer commercially in the future. You may have inquiries, especially for the pricing. And with this, you may contact us in Facebook, Agro-Technology Outreach Program, via email, ricd@anflocor.com, or you can directly call us at 082-234-711, local 893. And we are also active in Notion, in case you know Notion. It is where we update all our activities. You just search TDDS Laboratory. And of course, you may also contact us for possible collaborations and partnerships. I would like to end my presentation by showing you some of the operations in our laboratory. The determination of fungicide sensitivity is one of our major operations, with our continuous efforts to ensure high efficacy of the products that we apply commercially. And we also have microbiological laboratory, currently catering internal samples, which we plan to expand in the future. You know, the banana industry in the Philippines just recently embraced these technologies, and we are in the forefront in incorporating these tools to ensure the production of high-quality banana fruits. Our goal is to develop measures that are not only effective but are also operationally viable in the field, and hope to provide more impactful results for the banana industry. We truly have a vast potential for this, and we are even looking to expand our reach beyond pathogens, banana pathogens, in the near future. We do look forward to serving and collaborating with you all soon. Thank you for your attention.
Wow, thank you very much, Ma'am Bea. She's joining us from USA. Malamig naman dyan. Okay, I'm sure many of you are already wanted to ask questions, no? But please put your questions in the Q&A part of this Zoom meeting. For those in FB, you can type your questions in the comment section. Okay, from the banana industry to another banana businesswoman, let's proceed to our next speaker. Our next speaker is Dr. Remedios Plamiano. She is a Professor 6 At Mindanao State University, General Santos City, she finished her bachelor's degree of science in biology at Mindanao State University, her Master of Science in Microbiology at UP Diliman, and PhD in Horticulture at the University of Southeastern Philippines. She also received a certificate of teaching chemistry at Notre Dame of Marble University. She was the Science Department Chair at MSU Jensen from 2013 to 2017. She was the proponent for the establishment of molecular biology and microbiology laboratory at the Science Department College of Natural Sciences and Mathematics, MSU Jensen campus in 2013. She is also the program leader of the research project on development of microbial inoculum for sustainable rice production in Mindanao, funded by CHED in 2017. She is also the project leader of the MSU Microbiology Testing Center, funded by DA-BAR ASEF in September 2019. She is also the proponent of the MSU Banana Tissue Culture Laboratory by DA-BAR ASEF in July 2020. She is the proprietor of Greenland's Agritech Ventures, a banana tissue culture specialist, and a registered microbiologist. So to discuss commercialization of biotechnology applications for crops and ornamentals, please help me welcome Dr. Remedios Flamiano.
Hello everyone! Bago ako mag-umpisa ng aking talk, babatiin ko muna kayo ng isang napakagandang umaga at gusto ko ring pasalamatan ang Philippine Genome Center, ang faculty ng College of Science ng UP Mindanao for this opportunity to share my experience on how I was able to utilize biotechnology and help me build my profession and career in plant tissue culture. So for this year's PSBMB Regional Zoom Conference, I'm going to talk about commercialization of biotechnology applications for crops and ornamentals. So I am actually a professor and a researcher, and at this stage in my career, maybe I would consider myself as a trying hard entrepreneur who has accidentally landed into this field in the early 2000 after I was invited to join a team to produce banana tissue cultured seedlings for the growing banana industry in the Philippines. Banana tissue culture is a growing industry in the Philippines, and this is making use, yung pagpaparami ng mga tanim is making use of the plant tissue culture technology, which is actually a biotechnology. So to define plant tissue culture technology, it is a technique of growing cells, plant cells, tissues, organs, or the whole organisms in vitro using artificial culture medium under sterile conditions and of course controlled conditions. Now, yung plant tissue culture is a technique that has been around for more than 30 years already. And in fact, it is seen as an important technology for developing countries because it is used for the production of disease-free, high-quality planting materials and for rapid production of many uniform plants. So, yung mga plants that are propagated by tissue culture are palm oil, ang pineapple, banana, sweet potato, tomatoes, orchids, no? Yung mga ornamental plants, and even herbal plants. Yung tissue culture has actually a wide application in research kasi pwede tayong gumawa ng mga biotransformation studies, mga plant cell immobilization, mga plant genetic transformation studies using plant tissue culture. Now, in the industry, pwede rin natin magamit ang tissue culture in the production of valuable chemicals which are not possible by other chemical means. So we can also generate novel compounds from plants using tissue culture. So in agriculture, ang gamit ng tissue culture is on micropropagation, yung pagpaparami ng same na quality ng planting material. And ginagamit din ang tissue culture to do plant transformation studies kung gusto nating i-improve, for example, yung quality ng mga crops. And if you would like to produce secondary metabolite from plants, pwede nating gamitan yan, no, ng tissue culture. At kung gusto tayong makapag-discover ng mga disease-resistant plants, we can do this by plant tissue culture. Ano-anong mga tanim ang nati-tissue culture? And what are these plants that are commercially propagated, no? Yung merong commercial demands. In the Philippines, yung market ng bamboo in 2019 was at $72,102.4 million. And this is expected to reach over $98,757.9 million by 2026. So, you could just imagine the demand for bamboo kapag nag-flourish na itong industry na to, no? Another plant that can be propagated by tissue culture, in fact, very popular siya in Europe, is Paulonia species. Maraming species kasi to, yung Paulonia elongata, merong tomentosa, no? And ang pinaka-popular na tissue culture ngayon, no, in Europe is Paulonia elongata because this is one of the fastest growing trees, no, with high timber quality. The plant can be harvested in 5 years time at malaki yung applications niya in construction. In fact, ginagamit din yung wood niya to make ships, yung mga components din ng eroplano, some musical instruments, ginagamitan siya ng wood ng Paulonia. The plant can reach at a height of 20 meters and it can measure 35 centimeter in diameter. So it is actually a very potential agroforest crop because of its light and strong timber. During the pandemic, maraming tao ang nabaliw sa mga ornamental plants kasi yung mga tao, they find very relaxing when they grow plants and flowers. So there was huge demands for tissue culture planting materials, and yung mga dumarating sa Pilipinas, they are mainly coming from Thailand, like itong mga aglonema, philodendron, yung calathea, anthurium. Maraming mga tanim na dumating sa Pilipinas coming from Thailand. Orchids, tagal ng malaking industry ang orchids. In fact, mga 1980s pa, nandyan na yung orchids, and it is making use as well of the plant tissue culture technology to meet the global demands ng orchids. The major player ng orchid worldwide is Netherlands. They capture about 39.67% of the world market. Netherlands is followed by Thailand, Taiwan, Singapore, and New Zealand. At karamihan ng mga orchids na produced from these countries are mainly imported to Japan, UK, Italy, France, and the USA. Worldwide, lahat ng tao kumakain ng bananas. What is very encouraging is that Philippines is the second largest exporter of banana worldwide with over 4 million tons production in 2019. This means that there was a huge requirement for banana planting materials, no, during this time. Okay, globally, actually, ang commercial tissue culture started in the USA. So between 1986 and 1993, world production of plant tissue culture was increased by 50%. In 1993, plant tissue culture production was at 663 million plants. Now, 2 years later, plant tissue culture production has increased by 14%. Galing ito sa mga tissue culture planting materials from China. 2 years later, ang production ng tissue cultured planting materials nag-increase to 800 million plants. So of this volume of tissue cultured planting materials, less than 20% yung nanggaling sa mga countries like Africa, Asia, Latin America. So, kumbaga, ang Pilipinas, nandun tayo sa less than 20% no, ng 800 million plants na propagate by tissue culture. Now, ang projection ng plant tissue culture plants for 2020 to 2024 is that the global flower and ornamental markets will grow by $28.98 billion in 2024. Who are our major producers ng plant tissue culture? So, nangunguna dyan, of course, is Netherlands, followed by Japan, Italy, USA, Thailand, and other countries, siguro kasama dito ang Pilipinas, no, sa 14%. The tissue cultured planting materials are mainly exported by these countries. Of course, the Netherlands, who is the top producer, followed by Colombia, Italy, Israel, Spain, Kenya, and 18% from other countries. What about Asia? Nasaan ang Asia in terms of plant tissue culture? So in Asia, Thailand is the leader in tissue culture. Now with over 50 million plantlets production a year, and mainly orchids yung kanilang pinuproduce. Currently, umahabol ang India. In fact, based on record, they have produced over 500 million plantlets per year. And Taiwan is also in the scene. China, they are also into tissue culture. Malaysia, of course, including Philippines. Kasi ang Pilipinas, yung plant tissue culture technology is mainly used in the production of planting materials sa mga banana plantation, no, to control yung mga viral diseases in banana such as yung mga bunchy top at saka yung banana brack mosaic virus. The only way we can produce good quality disease-free materials is by way of tissue culture. What's the status of plant tissue culture in the Philippines? We made actually a survey in 2017. Meron akong isang estudyante, undergrad student, who made a survey on the different plant tissue culture laboratories. I think he surveyed 30. Of these labs that she surveyed, 64% are into banana. Karamihan ng nagdi-tissue culture, banana yung commodity na pinaparami nila. And second dyan yung abaca, which is 27%. Meron ding nagkukulture ng coconut at saka 5% lang ang ornamental. About banana updates in 2020, our banana production in the last quarter of 2020 was estimated at 2.4 million metric tons. And this is 0.4% higher than production in the last quarter of 2019, no, which was 2.39 million metric tons. Nakikita natin na malaking industry talaga ang banana that relies on plant tissue culture. And bananas in the Philippines are mainly produced in Mindanao, particularly the Davao region, Northern Mindanao region, at saka Soxargen. Of the different banana varieties that are being tissue cultured, ang pinakamarami dyan is Cavendish, now accounting for more than 60%, followed by Saba, 54.4%, and 21% ang Lakatan. The reason for this is that ang Cavendish kasi is a variety that is being exported worldwide. Yung Saba, ito yung for local consumption lang, at saka Lakatan. Although Lakatan, pumapasok na rin ito siya in the global market. This is an estimated gross sales from tissue cultured bananas planted in 2016. So you could just see the data is based on 2.6% increase in banana production in that year. So for Cavendish, commercial plantations were able to make use of more than 4 million planting materials ng Cavendish. So this is more or less amounting to 62,440,000 pesos. Now, for Saba, they have planted more than 4,700 planting materials, estimated at $65 2,987,000, and for Lakatan, they have produced over 2,831,868 with an estimated value of 39,646,152. So roughly, this is about 170 million gross sales in planting materials for that year. Based on record, our banana production is increasing each year from 0.4% to 3%. That also means that there's also an increase in planting material requirement for these plantations. Another commodity that is being tissue cultured in the Philippines and very promising crop is abaca or the Manila hemp. In fact, the Philippines' total revenue from abaca fiber exports in 2016 reached 25.78 million US dollars, and this was 33.9% higher in 2015. So ibig sabihin, nag-i-increase din ang ating abaca market. And with that, nangangailangan din of course ng malaking planting material. And abaca fiber market trends sa ngayon is estimated to increase by 2024 because of the increasing applications of abaca in electronics, in shipping lines, in making cars, aside from it being used for handicrafts and bags. So, tumataas yung requirement ng abaca. Also, there is an effort to produce more coconut planting materials. In fact, research on coconut plant tissue culture is given so much fund by the Department of Science and Technology, PICARD, and the OST is mobilizing the researchers from the Philippine Coconut Authority, the University of the Philippines in Los Baños, and in Mindanao, also in Bicol, and the Visayas State University to produce more coconut planting materials. So they make use of this somatic embryogenesis technology, or the CSET, which is the tool for the rapid mass production of superior genetic stocks of coconut using immature flowers, embryos, and the plumule, or the meristematic part of the coconut embryo. So, meron ding actually effort ang Department of Science and Technology to further boost our ornamental industry. Palabas sila ng fund to establish the Innovative Tissue Culture Laboratory for Ornamental Plants in Bulacan. This lab is expected to help our ornamental industry, and they are expecting this lab to open in the fourth quarter of 2021. Do you find commercial plant tissue culture an interesting venture and profitable? Is it possible to start with a very low budget? So, ano-ano yung mga requirements, basic requirements for you to be able to set up a plant tissue culture laboratory? So, una-una, you must have a plant tissue culture facility. Facility should include a preparation area, an area for inoculation, at saka for you to grow your tissues. Very important. Meron ka dapat mga equipment. Ang pinaka-minimum na requirement dyan is kailangan mo ng autoclave, digital balance, inoculating chamber, you must have a pH meter para ma-adjust mo yung culture medium mo, and oven to dry materials for tissue culture, and of course, you must have technical expertise. In my case, paano ko naumpisahan yung aking plant tissue culture laboratory? Wala naman akong malaking pera at that time, only that I have a little knowledge about growing plants under sterile conditions. So what I did, I invested some amount to set up a tissue culture lab. I converted our master's bedroom na mga about 15 square meters lang into trial laboratory. So this is my house at Sarangani Homes Phase 1 in General Santos City. This is just actually a 50 square meter house. Ito yung aking dalawang staff in 2010. I started my laboratory. Because of our limited resources, we were still able to come up with 5,000 to 10,000 Mary plants per month. Kulang-kulang pa talaga yung production namin. Kaya nakikita ko, there's really a very huge potential for banana tissue culture at that time. So ngayon, naghanap ako ng paraan, and when I went to the Department of Science and Technology in Region 12, may nakita akong funding support pala na pwedeng i-avail yung mga entrepreneur na katulad ko. So, through the DOST setup program, the Small Enterprise Technology and Upgrading Program, I was able to get funding support in the amount of 1.7 million pesos para ma-upgrade ko yung aking mga laboratory facilities. From the funding support I got from the Department of Science and Technology, we were able to increase our production from 5,000 to 100,000 Mary plants per month. You could just imagine the effect of intervention in our operation, and that made us acquire more customers and serve more clients, so that in 2017, we were able to acquire a 1 hectare area along Tupi National Highway and then construct this laboratory that can hold 100,000 to 200,000 Mary plants. Because of our significant growth, we were able to serve not only small farmers, but also commercial growers in Mindanao, including Del Monte Philippines, Musahamat Farms, Asia Pro, Safi Agro, at saka yung iba pang mga commercial plantations, and that we were awarded as the best DOST setup adapter in the region in 2018. Ito yung aming nursery in 2010 na 20,000 lang yung capacity niya, and in 2017 na-increase namin yung aming plant tissue culture nursery to 350,000 ang capacity niya. And with the intervention na ginawa ng DOST, we were also chosen as one of the 5 finalists in the DOST Setup 2018 competition. So, this is nationwide. Finally, nung July of 2018, we were selected as the best DOST setup adapter at the national level. So, as a way of serving the community, actually yung aming mga plant tissue culture, we just sell our produce at a very reasonable price na accessible to farmers. We also accept OJT students, and I also allow TC students who are interested to work on plant tissue culture to work in my laboratory. And hindi lang sa Mindanao State University, tumatanggap din ako ng mga estudyante coming from other institutions like for example, yung Sultan Kudarat State University at saka some private colleges. We're also holding trainings and seminars on plant tissue culture, if there's an invitation. Aside from that, sa mga farmers naman, nagbibigay kami ng free na technical assistance to local farmers para ma-improve nila yung kanilang cultural management on banana plantation. Sa ngayon, we have applied for another second round of financial support from the Department of Science and Technology with our future intentions to use the money to diversify our produce to abaca, bamboo, and some ornamental plants. Yan ang aming future direction sa ngayon. I'm very much grateful to DOST for the support. And of course, gusto ko ring pasalamatan ang Mindanao State University in General Santos City for the opportunity to learn skills and join scientific trainings. We would not reach this far without the support ng aking mga staff from Greenlands Agritech Ventures. In closing, I would like to thank everyone here listening to my talk, and I hope I was able to inspire you with this humble experience I have in my plant tissue culture journey. So maraming salamat sa inyong lahat sa walang sawang pakikinig ninyo sa aking life story. Thank you very much.
Wow, thank you very much, Doc Mimi. Yung inyo talagang love to love is very inspiring. So before we proceed to our open forum, let me acknowledge the presence of our participants from Baguio City, Bohol, Zamboanga, Digos, Panabo, Cotabato, Calabarzon, Quezon, Bukidnon, Jensan, and of course, Osamis, and of course from Davao City. So, okay pa ba? So are you ready for our open forum? So to facilitate our open forum, allow me to introduce our moderator, the Director of Philippine Genome Center Mindanao and a board member of PSBMB Mindanao chapter. Help me welcome Dr. Lair Ani Morao.
Good morning, everyone. Indeed, we are delighted to hear from our 4 speakers this morning on their programs and projects in their respective institutions. And I'm sure our participants are itching to ask their questions. We do have them now here live with us. So please do send in your questions through our Q&A chat box. So good morning, panels, our speakers. Good morning po. Just checking if your audio is working well. You can say hi.
Morning. Morning,
Good
everyone.
morning.
Good morning.
Good morning, everyone.
Good morning.
So we have here a series of questions. So I'm going to ask these questions to each of you. Let's start with the first question directed to Dr. Reggie. So there's a question here asking if you do accept requests for toxicity and inflammation analysis in your animal assay lab. And maybe I just connect with this with another question for undergrad CMU students po, will it be okay for us to also use the technology and machineries shown by you? And if yes po, will it be free or may bayad po ba? Dr. Reggie.
Good morning, everyone. It's my pleasure to join this webinar, and we are having this virtual tour, no? Places that we cannot visit right now, we are visiting virtually, and we are so happy to hear the updates from the different institutions. So for CMU, we are still working on our animal assay lab. It just opened this month. We just received our mice. So we're still validating our assays, but there is a definite plan in the future to receive toxicity and edema assays. We will just make sure that the consumables will be available and of course the personnel. But of course the priority are still the Tuklas Lunas Development Center researches. For our services on DNA barcoding or other assays, as of the moment, we are still working on our license to operate. And as of now, we are just still accepting CMU researchers, CMU students, and what has been approved is still the fee for equipment use, meaning to say you have to bring your own consumables and you would be assisted in the laboratory, but they can access all the equipment if they are available, of course, because always the researchers are the ones that are prioritized in using the equipment. But for CMU researchers and CMU students, they are welcome to use all the equipment. And if we have collaborations with other universities through MOU, for example, or MOA, then they could also access our equipment. That is so far how they could access the available equipment and services here in CMU. But of course, the SPAL, the Soil Plant Tissue Culture and Laboratory, has services to outside clients, and also we have
phytochemical analysis in the Natural Products Research and Development Center for outside clients, and microbiological testing at the College of Veterinary Medicine for water samples. So for details, of course, you can always email CMU for details. Thank you very much for your questions.
So thanks for that, Dr. Reggie. So in other words, if you want to use equipment belonging to the Tuklas Lunas lab, it would be best to approach that through a collaboration, right? For now. Okay, so thanks for clarifying that. Our next question is for Dr. Ed. So Dr. Ed, it says here, hi sir, I am a proud alumna here. So glad that USM is so busy with biotechnology. I would like to ask if you have plans or ongoing projects on Cavendish banana soma clonal identification.
Okay, good morning to everyone. Thank you for listening to the webinar. As that for last about 6 years ago was soma clonal variation analysis or studies on here. For that one is for the yield, for the texture, and so they are now having this for field trial. For Cavendish, we if it is a plan, it's part of the plan. But I think there are already some laboratories doing that one, especially addressing the issue of the resistance. One, possibly we could incorporate that one with also one of these.
Dr. Ed, parang choppy po yung signal ninyo. Dr. Ed, medyo choppy yata ang
yes, marinig. Allah, marinig mo ako?
Ngayon po, okay na.
Ito. Sige. Diretsuhan na lang. So, we have done it in Lakatan, and there are advanced lines now for field trial. And one of the future plan could be the Cavendish because of some problems now facing the Cavendish. Thank you.
Okay. Thank you for that very short, very quick answer, Dr. Ed. Okay, let's go to Ma'am Abea. Ma'am Abea, curious lang po ako. So you have this laboratory, let me just go back to that. It's called the Agrotechnology Outreach Program, right? It's a laboratory that provides lab services to
yes, ma'am.
Outsiders, to non-Tadeco. So sino po usually ang clients ninyo? For example, do you offer this to small farm, small holders also? And for example, are the services affordable enough for these farmers?
Yes, ma'am. We are offering po the services to outside clients. Our customers are largely on our small growers and those who wanted to avail our services. And the services, the cost or the prices are really geared na maging affordable siya for the growers, not just the corporate ones. Yun po.
Okay, thanks, Ma'am Abea. Now let's go to Dr. Remedios, Ma'am Mimi, Dr. Mimi. So my question po dito sa iyo, it says, not related to biochem po, pero we are currently doing a research paper regarding plant propagation using water propagation with the application of rooting hormones. We are undecided if we have to put nutrient supply to the water medium, for example, liquid fertilizer, since the rooting hormone that we will use already contains active ingredients to help the plant grow.
Okay. Ang problema ninyo is gusto lang ninyong initiate ng rooting. Did I get it right? kung totoo na routing lang, well, may mga studies na they can only incorporate yung mga hormones, like for example, IBA or kaya NAA, no, these are routing hormones. Pwede siyang ilagay lang sa tubig, no? Kung hindi masyadong maselan yung mga tanim. Pero ang experience ko with halimbawa tissue cultured siya, no, pagkatapos ilalagay ninyo siya sa routing na medium, kailangan meron pa ring nutrient yan kasi they will not develop good roots kapag walang nutrients. But for siguro mga simple lang na mga plants, no, pwede na yung simple na water with the rooting hormone. Basahin nyo lang kung ano yung range ng level ng hormone na applicable for that crop. Actually, marami kasing mga literatures na sa internet na pwede ninyong tingnan, no? Lalo na ngayon, 'di ba, marami tayong mga plantitos, plantitas. So common na mga tinatanong nila, how can they further improve yung propagation ng mga tanim? So ang advice ko dun sa mga mga entrepreneurs na mahilig sa mga ornamentals, they can try out itong mga available na mga rooting hormones. Actually, meron sa natin mga agricultural supply, may mga available dyan na rooting hormone, like for example NAA, no? So pwede niyong i-trial yan kasi medyo common yan na hormone, at saka IBA. So ang gagawin ninyo, kailangan talagang mag-trial, no? Kasi per crop iba-iba yung hormone range na kailangan nila. So dapat maging scientist din kayo kahit na papano. Okay? Thank you.
Sige po. Thank you po. We also have a question here again about many are interested on expanding your projects into services. So we have here a question for CMU or USM. It says, I want to ask if you are accepting requests for DNA barcoding analysis po. If so, how much per sample po? Dr. Reggie and Dr. Ed?
For DNA barcoding, I would suggest proceed to CMU because that's their specialty.
Okay. Dr. Reggie?
As of now, we don't have that services, but we have equipment use for you. So we just need to have collaboration, then some RAs to come here, bring the consumables, and we can assist them in their laboratory works for DNA barcoding. So far, ganyan pa. Wala pa kaming tao na talagang devoted to do the DNA barcoding. Thank you.
Thanks for that po. Related to services again, Ma'am Apea, may tanong dito. I would like to know, how do we diagnose FOXTR4 nowadays, maybe in your laboratory, and how much would it cost?
Okay, thank you for that question, Ma'am. We are using real-time PCR to detect TR4 in our lab, and I will get back to you with the cost or the pricing. You can send me an email or you can contact us for the pricing.
Yeah,
Thank
so
you.
maybe you can post your contact information later po sa chat. So
yes,
ma'am. Send their inquiries.
Thank you, Ma'am.
Thank you po. Okay. Kay Ma'am Mimi, siguro itong the other next question here. Do you cater MS students working on hybridization of ornamental aloe for mass production? Or maybe Dr. Ed can also chime in on this question.
Kung medyo merong research na gagawin, like for example, hybridization, mukhang hindi siya kakayanin sa laboratory ko kasi ang lab ko is only for mass production, no? Medyo kulang yung aming facilities to do some hybridization studies. So pagka merong component na mga hybridization, pwede yan siguro sa kay Dr. Barlaan. Pero kung meron na kayo, for example, na hybridize na, no, at saka sa tingin ninyo ang study na gagawin ninyo, i-propagate nyo na lang yung result ng hybridization na yon, pwede ako makatulong on that aspect.
Okay, Dr. Ed?
Okay. Ang tanong kasi niya is specific sa aloe. So we are not doing, although we are still starting this year yung different ornaments dito sa USM dahil ang North Cotabato especially makilala, they are the center for genetic diversity sa ornamentals, ang daming kuan. So we are establishing this year and we have not gone through a hybridization muna. Otherwise, kung ang hybridization aspects, pwede kami mag-cater dyan sa other ornament, sa mga commodities, mga food-based na mga commodities. Sa ganun, we could help them in that area.
Okay po. So again, you can post again your contact information in case they want to connect with you regarding this matter po in the chat.
Ma'am Lair,
Yes, po.
pwedeng mag-add ako sandali?
Sure.
For the information din ng ating listeners, meron kasi kaming isang faculty na returning from US. He was a Fulbright scholar, si Paolo Tagalugin. Babalik na siya sa aming university sa ngayon. He was working on hybridization, no? Improvement ng crop, no? Genetic transformation, something like that. I think the coming year, baka magkaroon kami ng studies kasi may lab na rin kami on plant tissue culture. So yung laboratory namin baka pwedeng makatulong sa mga thesis students natin to work something about plant propagation. So meron na tayong expertise who can help with you. Just coordinate with the Science Department of Mindanao State University sa mga needs ninyo on research at baka makatulong kami, no? Okay.
Okay, thanks for that, Dr. Mimi. So we have also many questions here. I think these are questions from students. Basically, I think what they want to know is, for example, I think they're really interested in doing some of the projects that you are working on. So for example, can they do OJT in your institutions? That's one question. And the other question, do you accept training for molecular techniques? So can we have a answer from each of you? Let's start with Dr. Reggie.
Good morning again. So for trainings on molecular techniques, actually we started that way back 2016, no? But of course, due to the pandemic, we canceled it, but we really are doing it supposed to be yearly. We are open for trainings, no? So we offer several trainings, not only on molecular techniques, even on mushroom culture, on fern spore culture, mga ganon, no, na mga trainings. You just have to link to us, no? Yun lang ba yung tanong? Okay.
Yes. Thanks, Dr. Reggie. Let's go to Dr. Ed.
Okay. Marinig na ako? Mahina pa rin?
Okay na.
Okay na. Okay, thank you. So dito naman sa USM, open naman kami for training, not only students, pati yung sa mga laboratories who want to have a training. Because of the pandemic, we need to schedule with a limited capacity. Pati sa tissue culture, sa molecular training, etc. So yun, limited. Ikuan mo na, contact us para mag-arrange because of the situation sa COVID pa ngayon. Lalo may lahat ng may mga bagong variants na ngayon. May Delta, Lambda, kung ano pang fraternity dyan, lumalabas.
Thanks for that, Dr. Ed. Ma'am Abea, sa inyo po, kasi you're in the industry, does TADECO accept OJT and do you provide trainings as well?
Yes, ma'am. It is different for the industry, no? Kasi we have samples to cater and everything. But for OJT, yes, we do accept, but not for now. Talagang nag-close kami for OJTs gawa nung pandemic. So we are very strict with those guidelines. And we do have trainings, but it will be on a collaborative manner. So as part sa industry, we really have to enter into a collaboration before we go with the training. So yun po. And we suggest also to, again, contact us because we are open really to collaborations. Okay. Thank you po.
Thank you. Dr. Mimi,
nabanggit ko na kanina, no, na as a way of my thank you, to the Department of Science and Technology. My laboratory is open for training. So we accept OJT, tapos even thesis students. So just like Dr. Barlaan, we need to schedule groups kung merong grupo na magti-training kasi in the lab, especially this is micropropagation, we cannot cater students na marami, no? So probably we can cater 2 or 3 at the maximum, no, at a time for training inside the laboratory. Pero kung ano siya, nursery management lang naman, pwede tayong mag-cater ng marami, no? But pero kailangan pa rin nating mag-schedule, no? Kasi nga pandemic ngayon, we don't want we're following quarantine protocols, no? So kindly send your letter na lang to us should you wish to get training on plant tissue culture, o kung inside ba yan sa laboratory or gusto niyo sa nursery management lang, no? So please state specifically your the training that you want to know specifically, no, on plant tissue culture hanggang sa nursery management. So may FB page kami, just please send your message there at Greenland's Agritech Ventures, and someone is answering queries from clients, even students, who would like to study. Okay.
Okay. Thanks for that, Dr. Mimi. So we have a lot of questions, but we only have limited time. So maybe as a last question for you guys, pang Miss Universe po ito na tanong. May tanong po kasi dito. So for example, we are currently in this pandemic right now, and we are experiencing a lot of problems, especially also in the agricultural industry. Most of you have projects on agriculture. And another thing in the agricultural industry is the fusarium problem. So maybe the question is, how can we use your projects, your initiatives to contribute to the solutions, to provide solutions for our current pandemic and also for the fusarium problem? Let's start with Dr. Reggie.
Okay, so thank you for your question, pang Miss Universe question. So, of course, with Tuklas Lunas, we are into anti-inflammatory products. We are currently on development. No? So we have functional foods, and eventually we would also venture into anti-infective screening, antiviral screening. So we hope to contribute to the pandemic through that. And if need be, we put up here a testing lab for RT-PCR. We are also willing to do that. So thank you very much.
Okay, thanks, Dr. Reggie. Dr. Ed?
Okay. Pang Mr. Universe ito, no? Okay. Okay.
Yes.
Okay. In the area, kasi tawag nito, how could we help? Una, sa banana industry, we have just developed technology using the PCR, real-time PCR, and the digital PCR, particularly in detecting the diseases in banana. Of course, may mga limitations ito siya, although qPCR is now used, but there are some discrepancies doon sa, lalo pag maliliit ang quantity ng one. Ang maganda sa digital PCR kasi, ma-quantify mo yung load, viral load, fungal load, bacterial load doon sa kwan. Kaya tulad na ipikita kanina is that this would help particularly in detecting, akala mo healthy, ikuanan pala, infected naman pala siya. So in other words, we will be pitching this technology for the banana industry, how we could help them, especially in forecasting the soil, because this technology can determine the disease in the soil, in the plant. Ganun na siya. So we can help them, what do you call this, especially yung area na produced, pati yung area for production, para at least merong preventive measures. Okay, that's for banana. And you have also for, ano pa ba, marami kasing crops. Ganito na, general na lang. Ganito, we have technologies dito na maitulong sa mga farmers, stakeholders, sa mga processors, sa cacao. Okay. One of the project din namin dito sa aming NICER, magbibigay kami ng libreng planting materials para mga sun nurseries. These are certified true to type na mga varieties para mag-propagate sila ng high-yielding true to type varieties with good quality. Magbibigay din kami ng mga planting materials sa communities para sila rin magsarili sila magtanim para makaproduce sila ng mga tableya for to yung sa health nila, mga antioxidants. So marami pa. So yun lang muna. Thank you.
Thank you. Pwede na po pang Mr. Universe. Next contestant, Ma'am Abea.
Hi, Ma'am. Thank you for that question. In terms of pandemic, paano kami nakatulong as an industry, a Cavendish producer na nag-export ng bananas? First is, of course, we have guidelines for our colleagues at work, field workers and everything. Hindi kami nag-stop ng operation, but we did have measures na continue pa din yung food basket ng aming mga kasama sa trabaho. In terms of ginagawa ng, for example, ng TADECO to address Fusarium wilt or Panama disease, we know that there is no single control for the disease. So we are doing researches such as prevention, how to prevent, how to eradicate, and of course, the most important, how to detect, because the correct diagnosis will come from a correct detection. So, and then, of course, what we would like really to advocate is that molecular, in my experience sa nasa industry, sometimes kasi we have at the back of my minds na we are a producer first, and yung molecular is mahirap siyang i-breach ang gap. But now we are trying to advocate na talagang tingnan yung details on how to really eradicate and not just on the general terms. How do we really efficiently apply and how do we really relate yung kunwari detection ng soil? Sinasabi ni Dr. Barlaan na sometimes ang farmers hindi nila nakikita yung disease because there is no plant and there is no symptoms. So those things na ginagawa natin ngayon, especially on limit of detection and paano siya nag-i-infect, it would really very much help the banana industry. But again, there's no really single solution to the problem. So biological, cultural, and everything, and especially discipline din ng mga workers. So yun po. Sana ako manalo sa question.
Yeah, but thank you to the TADECO for being for recognizing also the value of techniques in molecular biology and how we can use them in real world problems.
Yes,
all right,
ma'am.
let's move to Dr. Mimi.
Okay, since sa propagation I'm really strongly advocating na ang mga farmers natin should use tissue cultured planting materials if they are into banana production. Kasi malakihan ang tinatanim, and so if you happen to introduce something that is diseased already, it will contaminate all your plants and it will lead to failure in the production. So kung titingnan natin, mura na ang tissue culture ngayon compared sa suckers. Yung suckers, binibenta na rin ngayon tag 10 piso. No, but then you are never sure if these are already infected or carrier sila ng mga sakit. So mag-invest na kayo ng tissue cultured planting material na itatanim rather than sugal kayo sa hindi nyo sigurado. Kasi malakihan yung tinatanim natin. So we do not want contaminations to happen in the field and even spread of diseases. Kaya I strongly suggest go to accredited tissue culture laboratories and get your planting materials from there so that you are more or less assured of good harvest. Thank you very much.
Yan, salamat po sa ating mga speakers for sharing their insights and knowledge in this morning's open forum. So thank you very much, everyone. Now let me bring back to you, Lei, for our next batch of speakers.
Thank you, Lair, and to our first 4 speakers. Okay, so okay pa? Okay, so let's proceed to our 5th speaker. So our speaker is Assistant Professor Rovijem Villame. She received her Bachelor of Science in Food Technology from the University of the Philippines, Mindanao. She served as a research and development, quality assurance analyst, and technical assistant in various private food processing companies before she decided to pursue her graduate studies, where she finished her Master of Science in Food Science with a minor in microbiology at the University of the Philippines, Los Baños. She is currently serving as an educator and researcher at the UP Mindanao, and is part of the Philippine Genome Center Mindanao as project staff of the Omics Laboratory Services Division. Please help me welcome Ms. Assistant Professor Rovijem Villame.
Good morning once again, everyone. I am going to present some of the laboratory services that we offer at the Philippine Genome Center Mindanao. For this particular presentation, I am going to focus on the services offered by the Omics Laboratory Services, or OLS. So PGC Mindanao, or PGC-Min, is actually an expansion of the Philippine Genome Center headquarters that is stationed at UP Diliman, Quezon City. So we are actually one of the satellite facilities of PGC, and we are currently housed at UP Mindanao in Davao City. With the establishment of PGC-Min, Omics research is now within closer reach to the Mindanao community, and we intend to be a catalyst for growth and development in the island. Our institution has the following major objectives: to establish a genomics facility to support research advancements in Mindanao. Second is to capacitate the scientific workforce of Mindanao through collaborative research, science education and skills trainings, and hosting of researchers. And to facilitate genomic research programs in Mindanao in the areas of agriculture, food safety, quality and security, biodiversity, environmental management, health, and human diversity. Okay, currently, the PGC-Min has 4 major divisions. We have here the Research and Development Division, or R&D. We have the Bioinformatics Division. We have the Omics Laboratory Services, or OLS, and the Science Education and Training, or SET. So I am going to focus on the services of the OLS. At PGC-Min, we offer state-of-the-art facility and equipment for omics research, particularly in genomics research. So doon din po nanggaling yung term na omics. Okay? It's derived from a field of study in biology which usually ends in omics, such as genomics, yung study ng genome, or metabolomics, such as the study of the metabolome, or a set of metabolites that is present or produced by an organism or a cell. Proteomics, yung study of the proteome, or a set of proteins na present or produced by an organism or a cell. Or metagenomics, which is the study of microbial populations in a particular sample. At OLS, we offer various laboratory services para i-process at i-analyze yung various samples na iba-ibang origin. Pwedeng plant or animal origin, pwede ring bacteria or fungi. We use various molecular techniques and workflows para ma-achieve yon. So in the next slides, I will be presenting to you iba-ibang mga workflows as well as mga equipment na ginagamit namin sa lab. In a particular molecular biology research or omics research, usually may sinusunod tayo na general process workflow. It may vary from one research or experiment to another, depende doon sa sample mo. Okay? So, but usually it goes like this. So, if, for example, you have a sample, and then kailangan natin yan siyang i-prepare, i-homogenize, or kailangan natin i-extract pa yung kanyang DNA, RNA in order to get these target biomolecules. So for instance, we'd like to get its DNA, RNA, or a set of proteins. Okay? So may series of steps dito para ma-achieve natin itong mga target biomolecules na yan. So may it be homogenization, DNA extraction, RNA extraction. And then when we have these target biomolecules, you would like to know if present ba sila, are they intact, ano ba yung concentration nila. So, dyan papasok yung quality assurance and sample characterization. And then we get the results, and from the results, of course, ma-interpret na siya for application. So, saan yung role or saan pwedeng makatulong yung OLS, yung laboratory services ni PGCMIN. So we can offer services here sa sample preparation and processing, as well as the quality assurance and sample characterization. And we can also help in the interpretation of the results. For sample preparation, we have various equipment and workflows para matulungan kayo for to prepare your sample, to break open your sample or your cell, for instance, to get your target biomolecules. So, meron kaming tissue homogenization na workflow. We have bead male homogenizer for rapid tissue disruption using small beads, as well as a sonicator. So, andito po yan na mga equipment. We also offer nucleic acid and protein extraction workflows. So for nucleic acid, we can offer DNA extraction and RNA extraction. So for both methods, pwede kaming mag-offer ng either conventional or using commercial kits. So for traditional or conventional DNA extraction, we can use or we can apply CTAB, and trizol naman for RNA extraction. But if you want, pwede rin kaming mag-offer ng DNA or RNA extraction using spin column or yung mga nasa commercial kits. Okay, aside from nucleic acid extraction, we also offer protein extraction workflows. Okay, and not only yung manual protein extraction, we also offer high throughput DNA, RNA, protein cell isolation. So may equipment kami for that to extract 12 to 24 samples per batch. So, ibig sabihin na high throughput, mas maraming yield, mas efficient siya kasi mas marami ang samples na napoprocess at the same time. We also offer spectrophotometric analysis for nucleic acid concentration and purity. So, pwede nating tingnan kung kadami, ano yung concentration ng DNA, RNA na na-extract ninyo, and as well as the purity, or gaano ka-intact, or meron bang mga contaminants yung na-extract na DNA, RNA. We also offer spectrophotometric analysis for total protein quantification, applying the Bradford and Lowry assay. We also offer ELISA, or yung enzyme-linked immunosorbent assay, for the quantitative detection of target proteins, peptides, antibodies, and hormones using targeted analyte antibody interaction. Aside from proteins, we also offer reducing sugar analysis through DNS. So, through this method, pwede tayong mag-detect ng reducing sugars and other reducing molecules via interaction with DNS. So, gusto namin kasing i-maximize din yung aming equipment. No? So, not only proteins, but we can also analyze sugars. Okay, we also have a fluorometer for the detection of free or conjugated fluorescent compounds by light-sensitive diodes, which offers a higher degree of sensitivity and specificity than your spectro. Ito po ay qubit fluorometer. Ginagamit to siya kasi mas consider siya as more accurate than spectra kasi di ba usually ang mga DNA molecules ay nasa 260 nanometers, but other biomolecules also absorb light at that wavelength. So sa fluorometer kasi, especially ito yung qubit, gumagamit tayo ng fluorescent probe. So doon talaga siya mag-a-attach or mag-hybridize doon sa target biomolecules nyo at yun lang yung madedetect. So, kaya siya more sensitive and specific. So, we use fluorometry or the fluorometer for total DNA, RNA, or protein quantification. And of course, hindi mawawala yung polymerase chain reaction or PCR workflows. So, ito yung molecular technique which amplifies a target DNA para ma-detect natin if it is present or not in your sample. So we have two PCR workflows. We have the endpoint PCR for gene detection, and we also have the quantitative real-time PCR or QRT-PCR for transcript or gene detection. Pwede siya for gene expression and virus detection. So, yung QRT-PCR na meron kami ay yung Quant Studio. So, it's a similar equipment and workflow na ginagamit din for SARS-CoV-2. And ito rin yung equipment na ginagamit namin for the detection of African swine fever virus or ASFV. Okay, we also offer electrophoresis services for both genomic and protein workflows. So electrophoresis relies on the principle on the separation of your target biomolecules such as DNA, RNA, or your proteins by their molecular properties. Pwedeng molecular weight, isa-separate siya by weight, by size, by their charge, or their isoelectric point. So we have here agarose gel electrophoresis or age. So, gumagamit ito siya ng agarose as its polymer matrix para i-separate yung ating DNA, RNA, or PCR amplicons. Okay? Titingnan natin if present or absent yung target natin na gene or kung successful ba yung naging DNA extraction procedure natin. Titingnan ba kung intact or may smearing or na-degrade ba yung DNA during extraction, ganon. So, ito yung usually ginagamit for genomic workflows. We also have 1D gel electrophoresis. Pwede ito siyang gamitin for the detection or separation ng DNA, RNA, or your target protein. So, pwede siyang genomic and protein workflow. So, ano ba ang pagkakaiba niya sa AGE or yung agarose gel electrophoresis? First, magkaiba sila ng polymer matrix na ginagamit. So, sa AGE, agarose gel ang ginagamit. For 1D gel, ang ginagamit ay polyacrylamide gel. And second is yung orientation ng pag-pour ng gel. Pag agarose gel electrophoresis, horizontal yung ating setup. Pero pag sa 1D gel, patayo or vertical yung pag-pour ng gel. So, yun talaga yung kanyang difference. Yung Western blot technique naman is one of our protein workflows. So, if you'd like to detect a target protein, pwede namin i-offer ito, Western blot. So, for example, meron kang target protein. So, from a mixture of a protein sample, so isa-separate muna yan siya using a polyacrylamide gel, so similar sa SDS page na setup. And then, pag na-separate na siya, kumbaga ilalapatan siya ng membrane and then ita-transfer yung na-separate na mga proteins doon sa membrane mo electrophoretically pa din. And then may set of antibodies para ma-detect yung target protein mo. So ganun lang yung kaibahan niya doon sa 1D gel electrophoresis. So merong membrane na kumbaga ita-transfer mo after the separation, ita-transfer mo pa siya sa membrane and from there, i-detect siya using antibodies. Yung 2D gel electrophoresis naman, ang pagkakaiba niya lang sa 1D is that, syempre, two-dimensional lang. So, sa 1D is based on molecular weight lang yung separation. For 2D gel electrophoresis, dalawa. Yung first dimension, pwedeng by isoelectric point, and then second is molecular size naman yung separation. Kaya siya 2D or two-dimension. So, meron tayong I think 3 protein workflows, no, yung 1D, 2D, and Western blot technique. Okay, for the characterization and detection of single or multiple cell types by cell size and cell morphology or even cell counting, we also have a flow cytometer. Okay, yan. Aside from the laboratory services that I've mentioned earlier, we also offer equipment and facility rental. These are for research programs or institutions that have yet to procure their own equipment or nag-establish pa lang ng sarili nilang molecular biology laboratory or facility. So for equipment rental, pwede po kayong gumamit ng molecular biology-related laboratory equipment, and or bioinformatics equipment. So I already included here the services of the bioinformatics division since related naman yung mga services natin since the data that you get from the laboratory will also be processed no, and analyzed. And then for facility rental, you get full access to all molecular laboratory and or bioinformatics equipment at PGCMIN. However, this will also entail several prerequisites. So, bago kayo makagamit ng aming equipment and facility. Of course, we will also assist you and give you training or orientation para naman maging confident din kayo paggamit ng equipment and the facility. So, here, a GLP test or yung Good Laboratory Practices test, kailangan yung mag-take nun and you need to pass the GLP test. And you will also be given an orientation, a laboratory orientation, pwedeng orientation for a particular equipment, depende kung ano yung i-avail yung equipment as equipment rental, or overall orientation if you do or if you avail the facility rental. And pwede rin po ito na with or without reagents. So, pwede kayong mag-rent ng facility with reagents or pwede rin kayong mag-rent ng facility or equipment without reagents. So, yung mga consumable costs will be added na lang doon sa fees as needed. So, ganun po. The trainings are actually, I think, primarily sa ano ito siya, sa SET division, sa Science, Education, and Training division, under kay Ma'am Cyrus. However, nilagay ko na rin to dito yung trainings as one of our services sa OLS since most of our laboratory staff ay tumutulong or they also help facilitate sa mga trainings na ito. So, sila pa rin yung ginagawang resource person for these types of training, especially for the molecular biology methods in the laboratory. And sinama ko na rin dito yung bioinformatics trainings. Okay? So, this is mainly from the bioinformatics division. But also, most of our staff, some of our staff are also, kumbaga, they also help facilitate these trainings. So, yun, you can avail trainings for molecular biology methods if you want to capacitate your institution or yung mga technical staff ninyo, researchers ninyo, if you have a research. So, pwede kayong mag-avail ng ganitong type of training. Since the incidence and the onslaught of the African swine fever virus in the Davao region, there has been critical financial losses among its hog raisers. And during this time, especially last year, the PGCMIN has played a very important role in assisting and providing molecular diagnostics services for its detection. And this was made possible through the collaboration of the Department of Agriculture, Region 11, and HogFADI, which is an association of hog raisers in the Davao region, and which led to the accreditation of PGCMIN Laboratory, or the OLS, as one of the laboratory testing facilities for African swine fever virus, or ASFV, by the Bureau of Animal Industry, or TAI. And we hope that this will be a start, no, para magkaroon kami ng benchmark for potential diagnostic services in the future. I think this was taken last year during the pilot testing for ASFV testing. And this photo was taken just last Friday, August 6. Kakatapos lang ng virtual audit inspection by the DA BAI. So, ayan, my sigh of relief after the audit. And of course, I'd like to introduce our team. So aside from me and Dr. Dan Marie Del Mundo, headed by Dr. Lair Murao as our director, so this team is composed of very hardworking and brilliant scientists and researchers who played a very crucial role in the development of the laboratory as well as its services. We'd also like to recognize our past team members who also contributed a lot to the development of the laboratory as well as its protocols and SOPs. Thank you very much for listening.
Thank you very much, Ma'am Gigem. Our next speaker is also from PGC Mindanao. So Dr. Richie May Gamot, we fondly called her Chimi, is a faculty member of the Department of Mathematics, Physics, and Computer Science from Mindanao since 2001. She is also a faculty project staff of the Bioinformatics Division of PGC Min. She took her BS Applied Mathematics and MS Computer Science from the University of the Philippines. She earned her PhD in Scientific Computing from the University of Warwick, UK. Did I mention it correctly, Madam Chimi? And worked on modifying an existing nature-inspired optimization algorithm to fit the computational chemistry problem of optimizing atomic cluster structures. For years, her research interest focuses on nature-inspired methodologies, its variation and numerical methods to solve various scientific optimization problems across the fields of mathematics, biology, chemistry, and physics. She takes advantage of utilizing high-performance computing architecture to achieve productivity gains and scalable performance. Let's all welcome Dr. Richie May Gamot.
Thank you po, Ma'am Alayla. Thank you to PSBMB. Thank you to all the officers currently in this webinar. And I would like to really thank the group for this opportunity because I'd like to make it known that I rarely get to talk to gatherings whose audiences are mostly biologists. So having been part of PGC Mindanao has allowed me to mingle with this very nice group and to get to know the discipline even more. Again, thank you PSBMB for allowing me this opportunity to discuss the services of PGC Mindanao Bioinformatics team to this group. And I do hope that this presentation will allow for more collaborations. Thank you po. Here comes the presentation.
Hi again, this is Richie May Gamot, and in behalf of the Bioinformatics Division of PGC Mindanao, allow me to present the services of our team. Our division provides three major services. We provide trainings and internships, data processing and analysis, and access to our HPC facility. Under trainings and internships, our division has provided training for undergraduate students on the topics of Linux command line, Python programming, and sequence assembly. We have also recently participated in a pre-conference workshop for the Philippine Society of Microbiology, where it was attended by undergraduate and graduate students, as well as academicians and researchers. The topics included sequence quality control, sequence assembly and alignment, and evolutionary analysis. We can also customize training modules specifically for the needs of researchers. We can do this remotely or on a face-to-face setup as soon as protocols allow us. We have also conducted internships for high school students and undergraduate students from different academic institutions. BS Biology students from different state universities from Luzon joined the Bioinformatics Division this mid-year to assist us in improving the Bioinformatics Operations Manual. Furthermore, in collaboration with other divisions in PGC Mindanao, high school students were exposed to various topics and hands-on exercises including mathematical modeling, Sanger sequence processing, assembly, and phylogenetics. We also provide data processing and analytical services for clients wishing to outsource data analysis for their next generation sequencing data. PGC Mindanao offers services relating to genome and transcriptome assembly of sequences. Our division also offers services related to variant calling. In this service, from assembled sequences, we can identify single nucleotide polymorphisms and structural variations from next-generation sequencing data. Results from this analysis have medical and agricultural applications. We can also do bioinformatics analysis to associate specific genetic variations with particular diseases. In genome-wide association study, the genome is scanned to look for genetic markers that can be used to predict the presence of a disease. This identification will allow the researcher to understand how genes contribute to the disease and develop better prevention and treatment strategies. Scripts for visualization can also be generated for better graphical interpretation of the results. We can also do sequence similarity search, where it is possible to determine the identity of an unknown organism against a sequence database of known organisms. By statistically assessing how well database and query sequences match, one can infer homology and transfer information to the query sequence. We can also do evolutionary analysis of organisms, including an analysis on their spatial and temporal relationships. Here, we will be able to determine the diversity differences at a geographic level. We can also show how an organism connects with its ancestral origin and possible divergence that may occur in the future. We can also provide customized workflows or analysis for researchers who have a specific research question but who are not familiar with performing the bioinformatics workflow associated with their research problem. We can do customized workflows from doing quality assessment of their data up until performing variant calling and functional analysis on the assembled sequences. We can then proceed with evolutionary analysis on the resulting sequences, among others. These customized workflows may be implemented by our resident bioinformatician using our local workstation or submitted as jobs to a high-performance computing facility. In relation to customized workflows, PGC Mindanao Bioinformatics team participated in resolving the taxonomic classification of bat mitochondrial genome sequences from Malagos, Davao City. The Bioinformatics Division of PGC Mindanao was also involved in an analysis of wastewater samples at Mintal, Matina, and other selected barangays in Davao City to determine the presence of SARS-CoV-2 virus. In that investigation, we want to ask the question which SARS-CoV-2 variants can be detected in wastewater, hence are circulating in the community. We are also currently involved in the assembly of SARS-CoV-2 genome sequences from clinical samples provided by our partner COVID labs in various Mindanao regions. In this investigation, we are answering the question which SARS-CoV-2 variants are found in COVID-19 patients from different communities. Included in the list of our training topics is the module on Python programming and shell scripting. In the absence of user interfaces when working with the terminal or while using the HPC facility remotely, we offer a programming course on Python and Linux command line with shell scripting to allow the researchers a venue to automate the jobs with ease. We currently offer Python program and Introduction to Linux command line and Bash shell scripting to interested individuals. Those who are already familiar with using command line-based bioinformatics tools, we make available our high-performance computing servers for clients who wish to do their own data analysis. Our compute servers have 24 cores with 384 gigabytes of RAM. Our file server can also accommodate up to 72 terabytes of data. PGC Mindanao's compute servers and file server are being housed at the PGC Core Facility for Bioinformatics located at the Philippine Genome Center at UP Diliman. Software and databases for different analysis are available, including genome transcriptome and metagenome assembly, genome alignment, read mapping and variant calling, genome annotation, phylogenetic analysis, data visualization, statistical analysis, and many more. Additional software can be installed upon request free of charge. Access to an HPC facility would allow for a more worry-free bioinformatics analysis. As bioinformatics requires a huge amount of computing resources, some experiments are constrained by limitations brought about by low computer hardware specifications and outdated software versions of a researcher's local workstation. With more cores, memory, and storage, a researcher can focus more on increasing productivity and scalable performance. Command line-based bioinformatics tools are available including quality control, sequence assembly, sequence alignment, and phylogenetic analysis among others. Finally, the most important entity that our division has are our training staff who makes all of the bioinformatics services a reality. We have Jan, Dudes, Polo, Mitch, Christian, and yours truly. That's it for the bioinformatics services of PGC Mindanao. Here are the references and images used in this presentation. Pricing and details about our services not covered in this presentation may be inquired from this contact information. Thank you everyone for listening, and again, this is Richie May Gamot, and I wish everyone a nice day ahead.
Thank you very much, Ma'am Chimi. Is there anything else that you want to add before we proceed with our next speaker?
Mamaya na lang po siguro, ma'am, sa Q&A.
Okay, thank you, Ma'am Chimi. Okay, let's proceed to our next, but definitely not the last speaker for today. So to discuss the initiatives of DOST Region 11 toward the advancement of biochemistry, molecular biology, and biotechnology researches and services, please welcome back the Director of DOST 11, Dr. Anthony Sales. Good morning, sir.
Mayong buntag, I am Dr. Anthony Sales. I'm the Regional Director of the Department of Science and Technology, and I'll be discussing the initiatives of DOST towards the advancement of biochemistry, molecular biology, and biotechnology researches and services in Davao region. Before I proceed, I would like to express my gratitude to the organizers, to Dr. Annabel Novero, the president of PSBMB Mindanao, for the opportunity to present these initiatives. Here is the outline of my presentation. In DOST 11, we have developed three core competencies on R&D management, technology transfer, and S&T services. Aligned to these core competencies, I will be talking about the assistance we give to researchers through the Regional Health Research and Development Consortium 11, and then the One Expert program, and lastly, the services we offer under One Lab. As we all know, research has a huge role in the competitive edge or advantage of the country to boost its health economy in the local and export markets. A research agenda is essential to maximize the impact of investments and is regarded as a key in strengthening health research systems. The research priorities for Davao region comprise of 14 themes. However, I will give focus on the health research priorities where researchers in the field of biochemistry, molecular biology, and biotechnology can greatly contribute. Infectious diseases is one of the region's priority areas. Apart from COVID-19 related researches, this includes research topics on prevalent infectious diseases in the region, including dengue, TB, and HIV. Davao ranks 5th nationally among regions with the highest percentage of newly diagnosed HIV/AIDS cases. On nutrition, functional food, and food safety, we prioritize researches on nutritional interventions to improve the nutritional status of the population. Researchers here can specifically explore dietary supplementation, food preservation, and food fortification as research topics. On the topic of non-communicable diseases, the region failed to meet the national targets of risk factor prevalence for diabetes mellitus and overweight adults. The region also failed to meet the national target of mortality rate from cardiovascular diseases. Thus, research on risk factors leading to NCDs and evaluation of NCD programs should be considered by researchers to undertake. On traditional or herbal medicines and natural products, research efforts should aim to study traditional and herbal medicines from discovery to formulation. And on innovation and health technology, research on development of technology in hospitals to increase its availability is preferred for funding. Specific research topics include development of hospital equipment and Filipino-made biomedical devices. Those priority areas serve as a guide of RHRDC 11 in focusing its resources in terms of funding, support, and activities. The Regional Health Research and Development Consortium 11 is the regional arm of the Philippine Council for Health Research and Development. And here are the services of RHRDC 11. Under the Regional Research Fund, young researchers can get up to 1 million funding support. Interested researchers must check the National and Regional Unified Health Research Agenda to ensure that the desired research topic is a priority for funding support. RHRDC 11 also offers a mentorship program that aims to empower our young researchers, to encourage them to submit research proposals and to pursue health and health-related studies. With regards to training support, will depend on the identified need. Examples of these are trainings on research ethics, research publication, and proposal writing. RHRDC 11 also gives support and co-sponsoring services and activities of its member institutions such as the Madayao of San Pedro College and the webinar series organized by the Davao Colleges and Universities Network. DOST 11 partnered with PNP 11 and PLGU Davao de Oro in establishing the respective COVID-19 testing laboratories. Moreover, the department supported the SARS-CoV-2 molecular diagnostic training program of the Philippine Genome Center, Mindanao. Here is the list of ongoing projects under RRF. evaluation of the antibacterial properties of cacao theobroma, husk extracts, ectoparasites of avian species visiting selected fruit orchards of Davao City, detection of possible zoonosis, carotenoid extraction and characterization from Davao carrots as a potential pharmaceutical colorant, evaluation of Philippine mango pectin as tablet binder for formulated sambong tablet, and plant-mediated synthesis of silver nanoparticles using mangosteen, pericarp, and their antimicrobial potential. Apart from these projects, the DOST-11 is also supporting projects under the Science for Change program and projects funded by the DOST councils. The Science for Change program, or Science S4CP, has 4 components. One is the niche centers in the region, or NICER, for R&D, which aims to establish R&D centers in the regions to promote regional development. Another component is the R&D Leadership, or RD Lead program, which engages R&D experts to lead in strengthening the research capabilities of the higher education institutions, which engage experts with strong leadership, management, and innovative policy-making proficiencies to lead in strengthening the research capabilities of HEIs and RDIs. The third component is the collaborative research and development to leverage Philippine economy, or CRADLE program, which aims to create synergistic relationship between the academe and the industry with the goal of invigorating Philippine R&D. And lastly, the last component, the Business Innovation through Science and Technology, or BIST, for industry program, which aims to facilitate the acquisition of strategic and relevant technologies by Filipino companies for immediate incorporation in their R&D activities. At the bottom of the slide is the website if you want to know the schedule of the call of proposals, deadline, and for more information. Let me now move on to the One Expert portal. One Expert is an interactive web-based nationwide pool of science and technology experts. It is intended to improve access to experts and technologies, particularly by people living outside of the major urban area centers, where most researches and technical institutions are located. In the same manner, One Expert will likewise bring the services of any accredited expert located anywhere in the Philippines to clients that need S&T assistance. These are the procedures for accreditation of experts under One Expert portal. And these are the documentary requirements for individuals who wish to get accredited in the One Expert portal. The documentary requirements for consultancy firms wanting to get accredited in the One Expert portal are as follows. An accredited consultant of a DOST regional office shall have automatic accreditation by other DOST regional offices, which means that an expert in one region can be deployed to the other regions of the country to provide S&T services or interventions to those regions. But we also have grounds for suspension as shown on the slide. And an expert's accreditation may also be revoked for the following reasons. The procedure for the suspension and or revocation of the accreditation of an expert is as follows. But I hope that no expert in our region will be suspended or his accreditation revoked because of these reasons.
Meet the new One Expert. Redesigned and improved to cater your needs. Developed by the Department of Science and Technology, One Expert is a one-stop portal for your S&T needs and requests. Search for an expert. With a database of S&T experts, One Expert allows you to search them by name, location, expertise, or affiliation. Apply for consultancy or training online. One Expert is your online link to DOST's consultancy and training programs related to manufacturing productivity, food safety, agriculture and aquaculture productivity, and energy management. Learn from a series of free webinars from the comfort of your home. Brought to you by our lineup of experts, Expert O or Experts Online comes up with interesting and informative webinars exploring the field of science, technology, and innovation. Post your inquiries at One Expert Opportunities. Inquiries posted on this page are visible to all registered experts as well as DOST personnel nationwide. Get access to relevant science and technology-related materials and resources through DOST SCII's Publication Bridge and its online library, Starbucks. Download our mobile applications now. One Expert PH, One Expert Lite, and most importantly, One Chat. This messaging platform aims to connect clients to experts and DOST staff with ease. To enjoy all these features, create your One Expert account now at www.oneexpert.gov.ph. Be part of this growing science and technology community.
Now let me present to you the Regional Standards and Testing Laboratories, or RSTL Davao. RSTL Davao is a PNS ISO IEC 17025 accredited testing and calibration laboratory certified by the Philippine Accreditation Bureau. It is also accredited by the Food and Drug Administration to handle the testing of food products for product certification and licensing purposes. Other certifications of RSTL Davao include the following: EMBDNR, BIFAR, DOH for water testing, as a water testing laboratory. The micro lab has the following accredited scope of parameters. We analyze for food and beverages for the following parameters. Also drinking water and wastewater, food and water for microbiological parameters, and plant sanitation, monitoring, and evaluation. The chemistry lab has the following accredited scope of parameters. You might need these services in your researches. We also have established recently the Halal Verification Laboratory, which has the following accredited scope of parameters: porcine DNA detection, ethanol analysis. Other capabilities of the HBL include alcohol analysis in alcohol-based hand sanitizers, isopropyl analysis, methanol, and ethanol analysis. The Regional Metrology Laboratory has the following accredited scope of parameters. So if you want your laboratories, if you want to have your weighing scales, refrigerators, incubators in your laboratories calibrated, you can come to the Metrology Laboratory of RSTL Davao. RSTL Davao also offers the following services: shelf life testing for food and beverages, mentorship on ISO IEC 17025 accreditation, and R&D technical assistance, as well as referral of testing needs to capable laboratories within the One Lab network of DOST. So what is One Lab? OneLab is a network of laboratories anchored on an IT platform which broadens public access to testing and calibration services at a single touch point. Through the robust Unified Laboratory Information Management System, or ULIMS, the referral system of laboratory activities across government and private laboratories in the Philippines and even abroad, some parts of the world, in the ASEAN region, in Australia, and in the Middle East, was successfully completed. The One Lab network has provided the industry, the academe, the regulatory bodies, and private individuals a faster, easier, and reliable laboratory technology to meet the regulatory, statutory, and customer requirements. One lab can enable biochemistry, molecular biology, and biotechnology researchers to have their samples, or by having their samples tested to or by capable laboratories.
The one-stop laboratory services for global competitiveness, or OneLab, is an IT-based solution envisioned to broaden public access to testing services of all the member laboratories in the network of a single touchpoint, allowing for a seamless handling of samples from receiving, referral, transport, and analysis to the prompt delivery of calibration and testing reports. So, how does OneLab work? Step 1. Fill up the online request form through the customer portal, customer.onelab.ph. Step 2. The customer hands in the samples to the nearest OneLab member laboratory. The testing lab receives your sample, conducts the necessary tests or calibrations, and uploads the results to the receiving lab. OneLab gives you the option to track the status of your test requests with either web or Android. Just sit back and wait, and you'll get the results at the soonest time possible. Step 3. You can then get the test results of your sample by pickup, courier, email, or fax. With OneLab, customers are assured of timely, easily accessible, convenient, and available services. One laboratory worldwide. This is OneLab.
Thank you very much, Sir Tony, for your presentation. Now, I would like to call back our moderator, Dr. Lair Animbrao, for the open forum.
Yes, good morning once again. So can we request our speakers to be highlighted, spotlighted? So this morning, the second batch of presentations were kind of different in a way because these are institutions which offer different types of laboratory services and capacity building initiatives. So I guess we will have a different set of questions for this batch. So again, welcome to our speakers, Dr. Robbie Jem, Dr. Chimi, and Dr. Tony. So can we just try if your audio are working? Just say hello to everyone.
Hello. Morning po.
Good morning. All right, so we are looking forward again to hearing from you. There's a series of questions here in our Zoom chat as well as in FB. Who would like to hear from you, regarding the services that we offer. So let's start with the first question here. So it says, hello, PGC Min. I am working on insect terraries where we mass rear different insect species for assay of new molecules. What we encounter in the insect terraries is we observe diseases on the insect larvae. Question. Does can PGC Min cater identification of the pathogen causing this disease to our reared insects? So maybe we'll hear from Ikem.
Hello, good morning po. Thank you very much for that question. Upon consultation then with Ma'am Lair earlier regarding this question, it's possible po that we can identify the pathogens given kung meron na po kayong isolates and meron na kayong initial characterization. Kung meron na kayong idea kung ano yung mga possible identities ng pathogens na yon. But kung wala pa po talagang idea, parang pwede po siya exploratory type, pero magiging ano na siya, hindi na siya service, hindi na siya lab service. Kumbaga magiging research type na siya. So pwede pong mag-apply ng metagenomics para i-explore yung bacterial or yung microbial communities doon sa sample ninyo. But that would entail, kumbaga, exploratory research po siya. Yun po.
Okay, thank you, Gikem. Let's go to Ma'am Chimi. Dr. Chimi, we have many questions here, but maybe let's start with, for sequence assembly and analysis training, may we know the estimated duration for this type of training?
Okay,
And
thank you po.
can I add also?
Yes, ma'am.
There's another related question. So it says, what's the minimum computer specs in order to conduct bioinformatic analysis? And if they avail of the bioinformatic training, do they need to bring their own computing devices?
Okay. So definitely we do have services related to sequence assembly, no? In terms of the length of training time associated with sequence assembly, it depends upon the background of the trainees or the attendees. Kasi if the trainees would need background on introduction to using the command line interface, they will need to allot time for that one. But if they are already acquainted with introduction to Linux programming and a little bit of shell scripting, they can go directly to sequence assembly, and then we can probably use user interface tools, yung mga GUI, tinatawag natin GUI. So they don't really need to dive into the command line interface. So pwede tayong gumamit po ng ganyan. If we are using the GUI or the graphical user interfaces po, maybe we can allot half a day for sequence assembly lang po. Sequence alignment, I think sequence alignment yung question. So that's it. But if we need to add the introduction to Linux, if we are gearing towards using the command line, maybe we can have a full day for that one. Kasi we still need to do introduction to Linux, and maybe dadaan pa po tayo ng sequence, assembly, and alignment para po makuha talaga natin yung full package po ng training na yan. In terms of ano pa nga ba yun? I think there was a question. Their own okay. So actually, if protocols would allow us, meron po kaming mga computers, meron po kaming mga workstations sa UP Mindanao. We have right now 9 computers or 10 computers that we can use to cater for trainings po. If protocols will allow us, maybe in the near future, God willing po, we can have face-to-face training. But currently po, what we are doing is we do online trainings associated with these types of activity. Ginagawa lang po namin, since of course we cannot provide you with the machines, you should have your own machine to be able to participate in the hands-on training. Magiging lecture tapos hands-on siya. And in terms of the specifications po ng inyong computers, at least lang po siguro hindi siya Intel Pentium and Intel Celeron. Mga i-core devices, mga AMD processors will do. And then maybe we would at least want your computers to have at least 8 gigs siguro. That would be safe. 8 gigs of memory po. And a good chunk of your storage would be good. Meron po kami actually, if you are really interested in all our services, our first part of our service evaluation, training evaluation, is to actually send survey questionnaires to all the participants, and then we evaluate the specifications of the machines that you have before we say that you are capable of engaging in those training. Did I answer all the questions po, Ma'am Lai?
Yes, I think so. Thank you, Dr. Chimi. Let's go to Dr. Tony. Sir, there's a question here. Does DOST cater histopathology, blood serum analysis of avian species?
Mukhang malayo yan sa sa mga services na pinoprovide ng DOST 11. But let me check with the members of the One Lab network, which, as I presented, is a network of private and government laboratories all over the country, and even outside. We have members in Australia, Thailand, Vietnam, and even Saudi Arabia. So let me check from the members if they provide services related to what you are asking. But the website of One Lab is easily accessible in the internet. So you can check the services provided by the members of One Lab. Thank you.
So it's good that we have this One Lab system now where you can have networking with other labs. So in case, there are services like this which are not very common, then we can just ask other laboratories. Is there contact information, Dr. Sales, on the One Lab website where they can make requests like this?
Yes, all the contact information of the different laboratories that are members of the network are indicated there, including the list of services, including you can actually access online the results of laboratory tests conducted by the different laboratories. So it's a very user-friendly system that provides easy access to our industry, to the academe, and other users of the services of the One Lab.
Okay, so thank you.
So, Dr. Murao, perhaps this would be my cue to invite the laboratories who just presented earlier to be members of One Lab. So what we will do is we will send formal invitations. I saw earlier that CMU is ISO IEC certified or accredited already, as well as the laboratory of TEDECO. So we will ask the National Program Management Office of One Lab to send invitations to CMU as well as TEDECO to be members of the One Lab network. Thank you, Dr. Murao.
Yeah, so that's the beauty of this webinar, bringing different institutions together and forming collaboration. All right, let's go back to Gigem. So, Ma'am Gigem, there's a question here. If they want to avail of services in the laboratory, what are the steps and what are the costs of the equipment that you just mentioned in your talk?
Yun. Para ma-avail po yung mga laboratory services, especially for wet laboratory na mga activities, you can send us an email at pgc.upmin@up.edu or edu.ph. And then, istate lang po doon kung ano yung, kumbaga, gusto nyong services niya avail or kung ano yung mga samples ninyo at ano yung tests na gusto nyong gawin for that sample. And then, pwede na kayo namin bigyan ng quotation or fees. So, ganun lang po.
Okay, thank
Pwede
you.
po i-type po.
Thank you, Ma'am Gigem. Let's go to Ma'am Chimi. There are specific questions on different bioinformatic tools, no? So let me just pull them together in one question. So there's a question here on whether you do phylogenetic rebuilding and sequence alignment. If yes, what tools do you use? And do you offer these as services? A second related question is, do you also offer services on differential gene expression analysis? Do you also provide capacity building for institutions that wish to upgrade their bioinformatics units?
Okay, so the answer to those questions, I believe, would be yes to all, no? In terms of the tools, on the top of my head, yung aming phylogenetic analysis, yung evolutionary analysis namin, we usually use, our bioinformaticians usually use MEGA, and then we also use BEAST with BGEL to be able to generate those phylogenies. And in terms of the other question, ano pa nga ba yung other question? Differential gene analysis po ba yun? Yes, we do offer that service. We can do customized analysis, customized workflow associated with that, and we can also do training for that as well. And gusto ko lang siguro idagdag dun sa question in terms of the rates po siguro ma'am, if I may add, kasi para hindi ko makalimutan din. In terms of the rates associated with all our services, at least in the bioinformatics division, we do have different types of categories for a different type of research groups. So we have specific rate po for private sectors. Again, we also have another specific rate for non-UP government organizations. And then we also have another type or category for MGC, Mindanao Genomics Consortium po, and UP organizations. So I would also like to take this opportunity para mag-invite din po sa mga hindi pa po member ng MGC dito to contact us at pgc.upmin at up.edu.ph to know how to be a collaborative, to know how to be a collaborator under MGC para po malaki yung ating discount sa ating mga trainings and services.
Yeah, thanks for bringing up the Mindanao Genomics Consortium. Indeed, we would like to invite the different institutions here to be part of that consortium as well. Now we have a question on microbial analysis for Dr. Sales. Dr. Sales, does DOST offer isolation of microbes from environmental samples like soil and water? And there's a related question. Does DOST accommodate requests? I'm not sure what the question is all about here, but the question is, if DOST 11 is accommodating requests for the procurement of microbial culture. So please go ahead, sir.
Yes, I'll answer the second question first. We do have a small collection of microorganisms, but these are microorganisms that we use as references for in the analysis of pathogenic organisms in food and water. No, so the collection is limited to those microorganisms. We sell pure cultures of these organisms to researchers. Yes, you can make the request, and then just give us time. I think we need 3 to 5 days before we can serve the request because we have to prepare the cultures first. We need to prepare fresh cultures. On the second question on isolation, I think our microbiologists can also do that using our existing protocols. No? And again, this would be limited to the samples that we normally analyze in the microbiology lab, and this would be food as well as water samples. Thank you. Back to you, Dr. Murao.
Thank you for providing that information, Dr. Sales. Now let's go to another question for Ma'am Gigem. Probably Ma'am Gigem can answer this. Is your center actively involved also in sequencing the genome of suspected COVID-19 Delta and Alpha variants patients? So I think Jeannie gave some initial, initial talk about background on this. Maybe you can provide more information, Ma'am Gigem?
Sa ngayon po, we are not capable of doing kumbaga sequencing ng mga infectious diseases like COVID-19 or SARS-CoV-2. However, we can expand our sequencing kumbaga capabilities later on in collaboration or in kumbaga sa tulong ng PGC main sa Diliman. And siguro for COVID-19, pwede po under GECO, pero research work po siya. But we are not capable of handling live ng mga infectious samples dito po sa facility namin sa UP Mindanao sa PGC.
Okay, would you
I
like
hope
to
I answered the questions.
Si Ma'am Jimmy, do you want to add?
Yes po, add ko lang. So, although na-mention ko na to in one of the slides sa presentation, but I would like to reiterate na lang siguro that yes, we are involved in generating assembly of the sequences. No? So, as mentioned by Ma'am Gigem earlier, we don't have the capability muna to sequence at the moment, but we are looking forward into going into that direction na hopefully we'll be able to do from the sequencing to the assembly po later on. And as mentioned, we are also involved in what we call a GECO project, wherein we are trying to pull together the different samples. So they provide us the samples, and then at least the bioinformatics division generates the consensus sequence, and then we upload it to a database which we call REDCap, if some of you are familiar. And then what our collaborators do from I guess it's safe to say University of Glasgow, what they do is they pull together this information, get analysis on at least the regional direction nung spread of the virus. As of now, we are still on the development stage, uploading of these consensus sequences. Nagagather pa po kami from different partner diagnostic centers and medical institutions po. I think we do have around 5 or 6 collaborators at the moment. So I could not divulge any more information than that, but we do have that as of the moment.
Yeah, siguro i-clarify ko lang, when we say we're not capable yet of high throughput sequencing.
High throughput sequencing.
Yeah, because we don't have
Thank you for that, ma'am.
And to add lang po, we cannot process live viral samples dito sa laboratory.
Yeah, so right now we are only accepting extracted RNA as the starting material, not the live sample. Okay, I have a question for Dr. Sales. Actually, it's not the Q&A. It's my question, probably for the information, of our viewers here. Because BOST 11, as you have shown in your presentation, it really offers a lot of assistance, related to science and technology. So you have the lab services, but you also offer trainings, mentorship, and you also have research funding. So for example, if there are certain individuals or institutions here who would, need help in establishing their laboratories or projects on biochemistry, molecular biology, and biotechnology, how would they approach their regional BOST offices for help?
Thank you, Dr. Mura, for that question. Very simple, no? They just have to write a letter of intent, no? Indicating the requirements for capability building or institutional development requirements. We have programs which we call capability building program in another council, I think we shared, they call it institutional development program. So these two programs are intended to build capabilities in terms of facilities development or technical expertise development. No? So you can indicate in your proposal your requirements for human resources development and facilities development under these two programs. You just have to prepare the proposal. The links to this to the call for proposals, as well as the requirements for the proposal preparation, are indicated in the websites of PShared, PCARD, and PCHRD. So if it has something to do with health, then that's PCHRD. If it's industry, energy, and emerging technology, that's PShared. And for agriculture, aquatic, and resources, research and development, that would be PCARD. But also we have the central office lump sum budget that can provide funding for other concerns outside of the thematic areas covered by the 3 councils. And at the local level, at the regional office, we can also provide funding for your concerns. In fact, we have provided equipment, laboratory equipment to UP Mindanao and other HEIs in the region. So you just submit the proposal along with the letter of intent. If it's intended to us, then you submit the proposal. But if it's intended for funding by the councils and the central office, then you just submit to us a letter of intent, and then we will endorse your concern to the central office. Back to you, Dr. Lair.
Yeah, that's good news po. So we hope everyone who is listening here, and they're just starting to build their own labs and projects. You've heard from Dr. Sales on how you can also get help from DOST on those aspects. Okay, let's go back to Ma'am Gigem, and maybe also Ma'am Chimi can answer this question as well. So the question is, if the I think this question is for Ma'am Chimi. If the bioinformatics analysis is done by UP Min Services and this is to be published, how do you manage the authorship of these researches? Maybe Gigem can also share how it's done for the laboratory services as well.
Actually, I think both Ma'am Gigem and I consulted with our senior researchers under PGC Mindanao, and from it seemed like merong consensus that if the collaboration is from conceptualization up until the implementation of the methods, I believe aside from citing PGC Mindanao in the paper, I think there should be some sort of inclusion in the authorship. Okay, so I think that would be the safest answer that I can get. But maybe if we really dive into that collaboration, maybe we can talk more on that between the collaborators and each of the teams na involved in that project or in that collaboration, and then maybe we can talk about the authorship. But definitely, if PGC Mindanao, any of the teams that contributed into the analysis, there has to be a minimum of citation of PGC Mindanao in the paper po. For the authorship, that can be discussed further po siguro. So, Ma'am Gigem, do you have any addition?
I agree with Ma'am Shimi regarding authorship. I think depende po yan sa mapag-uusapan. And of course, meron tayong terms and conditions na kailangan mag-agree or parang may MOA muna before para clear na from the very beginning. So pag sinabi kasi namin na mag-avail ng service, yun yung basic services na pwede namin bigay, like yung mga laboratory services na yung staff ang gagawa, yung laboratory staff ang gagawa, pwedeng facility or equipment rental na kumbaga may payment. So meron din yan sa terms and references namin na pwedeng i-acknowledge nyo si PGC Min or i-cite siya or isama siya as authors. Depende po talaga sa mapag-uusapan. So it's really better na kumbaga makipag-usap muna sa amin and then i-clear muna kung ano yung mga objectives at saka ano yung kumbaga roles and responsibilities ng each collaborators para clear po siya. Yun lang po.
So in other words, we cannot generalize. It really depends on the parties. On the nature also of the work that is that will be done. But do you have for example, an NDA, non-disclosure agreement, just to protect, the work of the clients? Can anybody answer?
Yes po, ma'am. Meron po talagang NDA na kailangan para ma-protect din yung data na mag-generate from that.
Okay,
yes, I agree with Ma'am Gigem. Even in our trainings and internships, we do have an NDA for individuals. So I believe if it's a much more bigger collaboration such as projects, meron din po tayong NDA for that one.
Okay. So next question for Dr. Sales. It says here, I heard from one of the speakers earlier that BOST supports entrepreneurs in the line of biotechnology, such as Dr. Mimi in tissue culture or cacao. That's Dr. Ed, the cacao chocolate processing. Yeah, so for example, in the industry, does this program still exist? How can they apply? Yun po yung tanong.
Yes, the Small Enterprise Technology Upgrading Program or setup program is still ongoing. In Davao, if you're from Davao, you can just go to our office and prepare that letter of intent to avail of the S&T interventions that we provide under setup. And on that, based on that letter, we will conduct what we call a technology needs assessment or TNA to really ascertain ano yung kailangan mo doon sa negosyo mo. But this is intended for MSMEs only. So if you are a large enterprise, you are no longer qualified. But for MSMEs, you can avail of the innovation support. No, remember that for MSMEs to upgrade the quality of their products and to be more competitive, you need to innovate. And DOST provides both financial and technical assistance for you to implement innovations on the shop floor. So this would run from, or the range of financial assistance we provide is from 200,000 to 5 million pesos. That's the limit of my signing authority. So you can use these funds to buy equipment, to buy software kung kailangan mo, or to buy tools and instruments that you will use in your negosyo. No? So we will prepare a proposal, and based on this proposal, we can allocate the funds for upgrading of your facilities. Yes, this is still ongoing. Back to you, Dr. Lyer.
That's also another great news for our entrepreneurs here and those who want to engage also in entrepreneurship. Indeed, we have support also from our government through the OST. So do avail of this opportunity. All right, so it's almost time, but unfortunately we cannot address all the questions here in our chat box. So maybe just as a closing for our open forum today, my last question would be, since we're talking about biochemistry, molecular biology, and biotechnology, our dream is really to be able to see these technologies, these innovations, be translated into the community, especially here in Mindanao. So what do you think can be done to be able to reach, or to be able to reach that vision of seeing science being utilized in our community through your institutions? So let's start with Gigem.
Hello. Hello,
Sorry,
ma'am.
did you hear me, Gigem?
Can you please repeat, ma'am? Thank you. Hindi po siya na hindi po clear kanina. Opo.
So the question is, we really want to see science being used for the good of our community, especially here in Mindanao, where we have lots of challenges. So my question is, what can we do? How can we move forward through your institution in making this dream a reality?
Ay, pang Miss Universe ang question.
Hindi patalo. Kung yung first batch may ganong tanong, kayo din.
Siyempre kung sa tingin niyo po ay meron po kayong potential or capability to pursue research na sa tingin niyo ay maka-address ng research gaps, especially mga needs dito sa Mindanao. Kasi bihira lang po yung may mga omics research dito sa Mindanao. And through this institution, since meron na tayong facility and meron tayong equipment and meron din tayong mga collaborators na willing to help, lend their hand para sa technical expertise nila, andyan din yung support ng government, we should kumbaga push through and pursue kung ano yung mga research proposals natin. So I think yun lang po, and I'm also personally wishing na talagang ma-establish yung ating omics research dito sa Mindanao. Kasi marami pong potential na researches at saka marami pang pwedeng problems na localized dito sa atin na pwede natin i-address. Yun lang po.
Okay, thank you, Ma'am Igan. Let's go to Dr. Chini.
Thank you for that question. I think we have already been doing it, no, applying science into the community by virtue of doing these researches, these investigations on related to COVID-19. So as mentioned kaganina sa presentation po ng BioInfo, we have been participating in determining if there are presence of SARS-CoV-2 po na mga viruses sa virus po sa wastewater samples, and we are also determining po yung sort of biosurveillance analysis na rin po siya ng mga different cases sa Mindanao region. So I believe kahit yun pa lang po, we have already been doing the job in using science to provide something to our community. Maybe not really directly, but the information that will be generated from doing the workflows involved in these studies will be able to at least help siguro ating mga policymakers, no? If we generate a very good analysis at the end part of the workflow, they will be able to decide, let's say, intelligently po sa mga gagawin nilang different ECQs, barbecues, and whatever po sa ating community or sa ating localities po. So yun po, and then siguro idadagdag ko na rin po, there's no greater way of being involved in a community by collaborating with different institutions, research groups, and experts po. Yun po ang magsisimula po kasi doon yung ating generation of ideas, generation of research projects. So magsisimula po doon, collaborate with us, collaborate with different institutions, and definitely po later we'll be able to generate nice projects. So yun lang po. I thank you.
Thank you also for that, Dr. Chini. Let's hear from our DOST Regional Director, Dr. Sales po.
Thank you, Dr. Lair. Talaga pang Miss Universe itong mga sagot ng dalawa kong kasama. I'll try to top that para talaga ako manalo Miss Universe. Okay, tama yun, no, na that science should serve the people or serve the communities that we have here in Davao region. Actually, that's the mantra of the OST, science for the people. No, we should be able to bring science closer to the people kasi ultimately sila dapat ang mag-benefit sa results ng R&D natin, research and development. There are two strategies that we employ. First is, I'm sure you're familiar with the standing of the OST or of the Philippines in the innovation ranking. Medyo we're lagging behind other ASEAN countries in terms of GERD, gross expenditure for R&D, as well as the number of researchers, scientists, and engineers or RSEs. So how do we address this? DOST is now implementing the Science for Change program or S4CP, and under this program, we want to regionalize the funding for R&D. Kaya meron tayong NICER, we're trying to establish niche centers for R&D in the regions para mas mabilis ang pag-generate ng technologies, pag-generate ng R&D results na magagamit ng communities natin in the countryside. Kasi we want R&D to be inclusive, to be more people-centered or oriented, no? And then the second strategy is to generate more RSEs, more researchers, scientists, and engineers. How do we do this? Kanina nabanggit ko yung capability building program ng councils and DOST. So we want to train more people, more researchers. We want to mentor more researchers para makakontribute sila sa R&D kasi hindi counted kung if you're not actually conducting R&D dun sa survey, R&D survey natin. So I encourage our young researchers, our budding researchers to take advantage of these opportunities provided by the OST in terms of funding, in terms of training, in terms of other capability building activities. So sa tingin ko, Dr. Lair and our participants, yun yung strategies na pwede natin gawin na para makaangat tayo in terms of R&D. And more importantly, is the translation of R&D outputs into something that can be useful by communities and for them to establish what we call communities of practice. Kasi importante na hindi lang natin alam na merong siyensya, but we really apply science in everything that we do in our daily lives. No? So, importanteng ma-translate ang science into action. And we establish communities of practice down to the grassroots level. So yun siguro ang pang Miss Universe kong tanong. Sagot, Dr. Lair, back to you. Thank you.
Thank you for that, Dr. Sales. And I think it's really timely that we have a lot of young participants here in our webinar. I hope we take inspiration from what everyone said here. Kayo po ang future leaders ng ating bansa, and we hope that you could lead science towards being used in the community. All right, so that is our open forum for the second batch of speakers. Once again, thank you very much for taking your time to share your insights and knowledge to our viewers this morning. And at this point, I'd like to give back the floor to our host, Dr. Alayla.
Thank you very much, Lair, for those Miss Universe questions, and the winner is Director Anthony Sales. Thank you, Sir Tony. Okay, so now to present our certificate of appreciation to our speakers, may I call on Dr. Annabel Novero, President of PSBMB Mindanao Chapter, and Dr. Leslie Michelle Dalmasio. Good morning, Ma'am Les, National President of PSBMB, to award the certificate to our speakers. To all our speakers, please turn on your camera. So let me read the certificate of appreciation. The certificate is proudly presented to Dr. Reggie de la Cruz for being a resource speaker during the webinar Current Biochemistry, Molecular Biology, and Biotechnology Researches and Services in Mindanao on August 9, 2021, signed by Dr. Annabel Univero, President of the PSBMB Mindanao Chapter, and Dr. Leslie Michelle M. Dalmasio, National President of PSBMB. The same certificate is given to Dr. Edward Parlaan. And to Ms. Abea May Navasca. Thank you, Ma'am Bea. To Dr. Remedios Flamiano. To Assistant Professor Rovijem Villame. To Dr. Richie May Gamot. And to our director, Dr. Anthony Sales. Thank you. Thank you very much to our speakers. Please stay for a while for our photo op. Jans, we are ready.
Okay, ready?
So-
1, 2, 3, smile.
Okay, thank you very much also to our 371 participants. Thank you for staying. So thank you very much po to all our speakers for giving your time, for sharing your all of the services that are available in Mindanao. Okay, so before we proceed to the closing remarks and oath taking, I would like to present the officers of the PSBMB. Jans, can we? Okay, so sorry, we forgot about this. So we would like to present the PSBMB Mindanao Chapter 2021 officers and Board of Directors, led by our President, Dr. Annabel Novero, our Vice President, Dr. Reggie De La Cruz, yours truly, Alayla De Cadiz, Secretary, Dr. Remedios Flamiano, Treasurer, Dr. Edward Barlaan, our Auditor, Dr. Juliet Panghi, Dr. Lair Animurao, Dr. Cesar De Mayo, Dr. Abonawas Pendaliday, Dr. Gerardo Estoy Jr., Ms. Abea Nabasca, and our ex-official, Dr. Jaime Namukatka. Thank you everyone for making this webinar possible. Please let us give a round of applause to our 2021 officers and Board of Directors of PSBMB Mindanao Chapter. Okay, thank you very much. Now, to facilitate the oath-taking ceremony of the new members of PSBMB and to give a closing remarks, may I call on the National President of PSBMB. Let us welcome Dr. Leslie Michelle Dalmasio. So may I also request our new members to please turn on their camera. So we have Mr. Mac Arthur Cababan, Mr. Warren Caneos, Ms. Melanie Abatay, Mr. Ronaldo Bigsang, Dr. Lottie Kasim, and Mr. Jason Pukot. Can we turn on their camera and oath taking for them to follow?
Okay, are you all seen now? And okay, so may I request the new members to please raise your right hand and repeat after me. I, please state your name. As member of the Philippine Society of Biochemistry and Molecular Biology, do hereby submit myself to be governed by its constitution, bylaws and duties therein, and to uphold the ideals and principles of the society. So help me God.
So help me God.
Congratulations to our new members and welcome to the Philippine Society for Biochemistry and Molecular Biology. So we hope to see you in future activities of PSBMB.
Thank you po.
Congratulations po.
Thank you po, Doc.
Thank you, Ma'am Leslie. Can we take a photo with the new members?
Okay.
Jans. We'd also to remind the participants to get your certificate of attendance from this webinar, click the link in the chat box sent to you just now and fill up the evaluation form. Okay, Jans, we are ready for the picture
Okay.
taking.
Okay, ready? 1, 2, 3, smile.
Okay, thank you very much po, and welcome to the PSBMB family. For those who wanted to be a member of PSBMB, we will be posting the links for the link for the membership. It will be posted it is posted already in the PSBMB Mindanao website. Okay, so Ma'am Leslie, I will now give the floor to you for the closing remarks.
So it has been a very productive morning, I think, for all of us. So it's still not yet noon. So I would like to extend good morning still to DOST 11 Director Dr. Anthony Sales, to the PSBMB Mindanao Chapter President Dr. Annabel Novero, to the Board of Directors of PSBMB Mindanao, to the esteemed speakers of the webinar, and to everyone. I and the PSBMB National BOD are truly delighted that amidst the limitations the current pandemic imposes, the chapter is able to organize and hold this very relevant webinar for stakeholders in Mindanao. Perhaps it is the opportunity of a virtual gathering that made this activity more feasible and accessible, but nonetheless, it is a truly worthwhile endeavor at the right time. Your webinar shows and proves the research capabilities and the rich research opportunities in Mindanao. But more importantly, it promotes scientific pursuits, collaboration, and harmonization of efforts. The current pandemic also opens new research avenues, but biochemistry, molecular biology, and biotechnology remain at the forefront as we continue to battle emerging infections, not only through vaccination, but also through drug discovery, functional food development, and environmental protection and monitoring. Biodiversity protection and research also contribute to sustainability efforts and prevention of emerging infectious agents. And in Mindanao, you have rich biodiversity to explore and utilize. You are fortunate to have a very supportive DOST regional office and facilities that will address research needs. May this translate to more research proposals in and from the region. However, may there also be hand-holding of young and budding researchers so that there will be sustained efforts with long-term impact. So congratulations and thank you to the PSBMB Mindanao chapter for this webinar. We are looking forward to other activities that will emanate from this webinar, as well as other activities for the PSBMB members. Makabuluhang agham sa inyong lahat.
Thank you very much, Ma'am Leslie. May we request a photo opportunity with our speakers and also the officers of PSBMB Mindanao. Photo op po tayo with Ma'am Leslie, please. May I request other officers to please turn on their cameras. Andiyan na ba, Jans? Natataranta pa yung aming technical team. So before we end this, I would like to thank our technical team, PGC Mindanao, for helping us in this PSBMB webinar. We would like to promote also the media pages or Facebook page of PSBMB Mindanao and PSBMB National, and also the PGC Mindanao website. You can also check the future activities, especially the conference this coming December of the PSBMB National. We also have bioinformatics workshop and webinar also, and the third Mindanao-wide genomic seminar this upcoming October. These are all in the videos which will be played later on. So are we ready now, Ma'am Jans, for the photo op? Ma'am Bea, picture
I-on
na
yung
lang daw
amin,
po.
hindi kami nalaw.
Okay. Si Sir Ed daw, hindi mo analaw.
Okay, are we all ready?
Okay na, Sir Ed?
Okay, sige. Ready? 1, 2, 3, smile.
Next page ba?
Okay, Waki. Ready?
Waki.
1, 2, 3, Waki. Smile. Okay.
Okay, thank you very much. Thank you very much, everyone. So I am Alayla, and this is webinar, Current Biochemistry, Molecular Biology, and Biotechnology Researches and Services in Mindanao. Good afternoon and stay safe. Thank you very much, everyone. Yay. Thank you po.
Congrats to everyone. Congratulations, PSBMB.
Thank you, Ma'am Leslie.
And thank you to all the speakers. Very productive morning.
Thank you, Ma'am Les. Thank you to all our participants for staying. And to our new members, congratulations po.
Thank you, Ma'am Laila.
Anna, do you have anything else to say pa po? Okay na po.
Congratulations, everyone.
Thank you, everybody.
PGC,
Congratulations.
PSBMB.
I think we had 378, no? Jan?
Wow.
Yes, ma'am.
Sa FB, ilan yung ating, yung ating...
How about the participants? They might want to have their pictures taken.
Okay, ma'am. Sige, ma'am.
Oo, kasi...
Jan, yung mga... Yung participants daw, Jan. Jans, can you turn on the... umalis na ibang officer. Kami ibang participants. I think
About the off po?
Oh, yeah. Gusto
Kasi
niyo
pag gusto nila.
Okay, sige po.
Yan, sige. Yung ibang participants po, you may want to turn on your cam. Baka may data privacy issue. Meron pa ba, Jan?
Yung gusto join us.
Yeah.
Nandiyan pa sila.
Okay, everyone, please turn on your camera.
Yung participants, wala?
We still have 305 participants.
O yung team din daw, bakit naka-off? Pagsuklay na.
Na ang signal.
Picture na dyan.
Sige, ma'am.
Wala kang access.
Baka automatic on na lang lahat.
Yeah. Nice lagay mong hair, Madam Marian.
Oo.
Ito lang ha.
Ganda ang hair ni Lyre.
Si Ma'am Larden. Si Ma'am Larden. Sorry, ma'am.
Nag-alis na itong iba. Hi, Ma'am Lottie. Nice to meet you po.
Hello, Ma'am Lai.
Kinukwento po kayo sa amin ni
Open yung mga
Aaron.
participants kasi webinar.
Ongoing po yung ano namin ngayon, yung aming monitoring.
Disable ang task ng mga participants.
Kaya kaliwaan yung laptop.
Dapat mag-end sa.
Hindi
Yes, we cannot,
maka-access.
our
Yeah,
participants
sorry.
can't open their cameras po.
Unless
Only the
isa-isa sila
panel.
Hi,
i-open ng
Ronald. Big
Oo
son.
nga. Sige, tayo na lang
Congratulations.
muna.
Sige, kayo na lang muna, ma'am.
Hindi siya pwede ma-allow lahat.
Hindi,
Pwede
kailangan daw
isa-isahin mo.
mag-exit.
Okay. Nakaset kasi
Sige.
siya na no videos.
Yeah. Sige, tayo na lang muna mag-picture. Go, Jans.
Okay, sige. Ready, 1, 2, 3, smile.
Okay, thank you po. Hindi, gusto talaga hindi rin maka-access yung iba. Bye-bye. Bye-bye po.
Thank you.
Good morning, good night. Good morning, good night na ba dyan, Ma'am Rhea?
Nakangiti daw yung mga participants kahit wala silang video. Thank you. Thank you po. It's the thought that counts.
Salamat po. Hey, congratulations, guys.
Sige. Hindi ko alam. Comment. Naka-on ka.